Good morning everybody and welcome to dive 414 with the ROV SuBastian. We're on RV Falkor here at Ashmore Reef on the northwest shelf of Australia. This morning we have deployed the ROV in about 50 metres of water, and to start the day off, as we have been doing every day for this Australian Mesophotic Corals expedition, we are going to run a transect. That transect's going to travel across 300 metres of the seafloor, and we are just going to be recording imagery as we go along. We use that imagery later on back in the lab to start to quantify the diversity and the abundance of all the benthic invertebrates that are living on the seafloor. Really looking forward to today's dive because we've identified a very interesting looking reef area on the multi-beam data, and that's where we're going to be working around today. So you can just see on the screen now we're coming up to some good hard ground, and we've got our fingers crossed to see lots of corals today. I can see at least 3 in the view already, and so it's looking promising right from the beginning. Once we've done the transects, we're going to be focusing on collecting lots of coral samples for our population genetics study, and then generally collecting and looking for all sorts of organisms for the West Australian Museum. I'm Dr. Karen Miller. I'm going to be taking you through the dive today alongside Dr. Nerida Wilson from the West Australian Museum. So I hope you enjoy today's dive, and fingers crossed for lots of corals.
Just going to let the ship start moving first.
That's all right. This is quite a nice spot just to sit and gaze at the beauty. I can see one coral right there, dead centre. Oh, that's a good idea. Thanks, Russ. As always, first thing in the morning, we seem to have a little bit more current than we'd prefer, but typically it drops off through the day. So just getting the ship in the right position. We'll just have a little look around while we're here. Wow, just colour everywhere. That's that little chap. Pretty much every shade of pink imaginable in that photo. Could you just take that photo because it's not pretty?
Oh, right outside, so that's not well.
At least the water's nice and clear this morning. Yesterday morning we were confronted by a snowstorm by the time we got to the bottom, but that was in much deeper water. So nice to have good clarity. Probably should say relatively good clarity. There is still quite a lot of sediment in the water, but it's still manageable to get some good images. Oh, there's a little coral there. Well, is the boat in position, or are we still waiting?
Just give her another couple of minutes.
Another few minutes, that's all good.
Just to let him catch up. Dog needs rolling in and out. Okay, we can start moving slowly if that's okay.
Yeah, ready when you are.
Lift off. Okay, this is actually the start point here if you want to. We're going to have to go up and over the top like that. If that's okay.
That's the plan.
So if you want to press the start of transect now, I can start that.
Okay. Start transect.
Karen, if you just bump the camera a little bit to your left, we'll straight up there.
Bump the camera to my left. Oh, you've done it. Cool.
Yeah, I did it automatically. So, smart cam.
I didn't think there was possible to have more shades of pink, but I think we do.
Full palette, yeah.
Well, if the first 2 minutes of this dive are anything to go by, it's going to be a pretty nice day.
Let's try Angel first.
Hey, Jason, Adam here. Are we able to slow that move down a little bit? Yeah, no worries. Cheers, thanks.
That's 2 meters. fish here, eh?
It's teeming with life. And pleasingly, quite a few hard corals. Our mission out here at Ashmore Reef is to really just understand all the species diversity that is living in the Mesophotic zone. Ashmore Reef is a really isolated and iconic reef. It's actually closer to Timor than it is to Australia. It's right out on the edge of the Australian continental shelf. It's actually a marine park, and so it is well looked after, but we still know very little about the inaccessible deeper parts of the reef, and so that's our mission while we're here for the 14 days of the Mesophotic Coral Expedition, is to learn as much as we can about the Mesophotic zone, and all that information is going to be invaluable for helping the Parks Australia manage and preserve this reef area. This area is certainly dominated by lots of the octocorals or soft corals, huge diversity of Gorgonian sea fans and various types of soft corals, all these different beautiful pinks and oranges. Just spectacular.
You give me a favour and swing that boat around the ground and jump in this. Just gets out of the road.
Depth control.
Yeah, so
Hey Ben, I got an eye on the floats here if you want to move that by team.
I already done good.
Cheers.
Yep.
It's 600 with this thing.
He's alright.
Nice. Sorry, he's already had about 3 pancakes.
I'm enjoying your sunrise, Adam.
Take a mental picture for me.
Yeah, is there some way we can shift that deck camera around so we can all see the sunrise? Not quite. The weather here at Ashmore Reef is just spectacular at the moment. There's hardly any wind and the sea is very calm, so up on deck it's just spectacular. Last night we had a gorgeous sunset and it looks like the sunrise is going to be just as good. We of course are stuck in the bows of the boat with about 18 TV monitors in front of us watching all the various control aspects of the ROV and of course the imagery. So we don't get to see too much daylight, but the opportunity to see all this marine life is definitely worth it. can see at least 10 hard corals in that frame. That makes me very happy.
Point of interest for that one, perhaps?
Oh, that's a good point. Thank you. Yeah. Point of interest.
I'll just hover here and then we can have that as a come back and forage later.
That's correct.
What do you want to name that one?
Lots of corals.
LOC.
Coral party. How's that?
Alright. So you can eat and
Maybe bumped by a large shark or a large fish or something, or even knocked about in the current, or a cyclone may have had those effects, I guess. It looks like it was damaged a long time ago. It looks relatively well healed. Oh, look at those beautiful corals there. Little pancake looking guys are the ones that we'll be targeting later for our population genetics study. Just noticed a question there from Mary Alice Clark on the Facebook feed about whether the pink bowls are sponges, and the answer is yes, they absolutely are. Big, beautiful sponges. Sponges are often quite a dominant member of the mesophotic communities.
Bridge, ROV.
We've been seeing some really stunning ones here at Ashmore Reef.
Yeah, go ahead. Point 2, point 3.
Perfect, thanks, Jason.
might want to shut that. We might have them so you can- Honestly, so because it's cold days. So guys, a little look around here until the ship catch up a little bit.
All good.
So what we'll do is I'll find something that looks pretty interesting. We have a 2-minute break.
Oh, that's beautiful. That's a nice spot to rest. We were talking about the Christmas card photo yesterday, but I think this is quite a good one too, actually. It's very celebratory. Thanks, Russ. Oh, no, I can't zoom out anymore. That's cool. Christmas card photo.
We're going crazy on the higher res screen grabs over here as well, so desktop backgrounds.
Oops, it's not going to focus. Oh, there we go.
There we go.
You can see the detail of that soft coral. It's just gorgeous. All the polyps out feeding.
It's good.
It's easier to get into.
Okay, we're ship's good. Catching up there, so
Good to go.
good to go now. It's like something at Alice in Wonderland there. It's like, it's like these.
Having a tea party.
Exactly, that's what I'm saying.
Oh yeah, and there's all those ones in the background that is doing the tea party thing again.
There's one of you, if you.
Ooh, can we
Yeah.
yeah, point of interest that coral, please. It looks like one of the,
See this one ahead is?
this one here.
This is going to be,
Did you point of interest that coral? Thank you. That might be one of the.
What would you like to call that, Karen?
Call it a Pachy, P-A-C-H-Y, with a question mark. It's one of the ones that we are hoping to get for one of the student projects here. She's looking at ecophysiology, so trying to understand about the coral's metabolism and its feeding and its reproduction.
is that another one there, right in the center screen?
No, that one's the kind of filia-like one that we keep seeing. Oh, that sponge is huge. It's like a bath. That's not a teacup, it's a bath.
It's a big cup of tea, though. is a twofer. This one over here is a twofer too. Do you like lasers
Oh,
on that
wow.
there? Oh.
Ah, yes, lasers on. Thank you, John. No, yes, it is John there. Yeah. I just. I'm sure last time I looked it was James. I just got distracted right from the beginning with all that pink and all the incredible marine life. Easy to do. Interesting how we've moved from a field of sponges to now we're in a field of sea fans. In the space of a few meters. Oh, there's just a stingray in the background. Can you see him?
Yeah.
Just cruising through. Oh, he's coming to say hello. Oh, no, decided not to. No, he's a bit too far. Oh no,
Yeah, he's definitely not
bit
much.
fast for us.
Stir up the bottom.
He's just going to taunt us by just swimming in front of us.
Yeah,
Just
he's-
too far away to see properly.
He's quite noisy, so he probably doesn't want to come to you.
Yeah. Oh, do you want a point of interest, that sea cucumber?
Yes, please.
Because I suspect Nerida would like to come and look at it for parasites. The current's dropping right off.
Oh, that's nice. Nice. Sea cucumber. All right.
Bridge, ROV. Updating the waypoint color. Is that a good color for you? See that better? Oh, you got black. Okay, how about purple? Alright?
From that, it's the same colour as every. What you do is change the ring colour on it so we know which one's what. Just let it. Just put like a blue ring on it. Yeah.
We'll do the water collection halfway again as usual.
Waypoint 3's are
Yeah.
50 metre mark.
Yeah,
and we can
perfect.
Stop.
Good morning, Nerida.
That's what I figured about that.
We might waypoint that sea cucumber too and come back to it later and have a good look. It's very beautiful here today.
As soon as I posted the link on Facebook, people were saying, "Oh, it's a floral party." And I'm like, nice features because they're not too steep and crazy. Yeah, beautiful.
Yep, seen lots of hard corals. Tagged a few sea cucumbers to come back and look at.
Good fans.
This building's not too bad, but the current's dropping.
For enough days to rain now.
Oh, it was 1.3 knots on the surface when we started. But once we got down, it was fine, but it's been dropping ever since. So I think just we're starting to get into the thick of the tide. So I think it's going to be a good day. Touch wood.
Not much to do, I suppose, actually.
Diversity is amazing.
Housing thing.
Not up to the Christmas yet.
No, but that's the next waypoint is the Niskin one, so should be fine.
Guys, yeah. Got me over there.
That's mine, it's still in the office.
Center screen, is that the one that you're looking for?
You mean this?
No, it's sponging out.
Yeah. It's a very interesting shaped sponge. Kind of looks like one of those big bowl sponges, but tipped over and squashed, so to speak. Right, I shall hand over to Nerida for a little while.
We got this. some I don't know what photogrammetry, to be honest. But yeah, I've marked one thing there that was, look, this looks like it's pixels, but yeah, so see how you go. But as a minimum, you can just start with population from the next one.
Yeah, and that shallow practice there, are we on the shallow side of that?
We're kind of if I think we might get a little shallow there. So it just gradually goes up.
Okay, so the end of the transect down higher.
No, we go back down at the end of the transect. So
Okay, we'll try and pull that to that transect area.
yeah, if you can see some. Thank you. Thanks.
See ya.
So we will come to waypoint 3, which are this water sampling.
Cool.
Yeah,
Yeah.
very shortly.
Yeah, no worries. Good morning, everyone. This is Dr. Nerida Wilson from the Western Australian Museum. Pleased to be here again this morning in this gorgeous place. You've been probably watching it a little bit longer than I have, but I'm super impressed with these big fans and big sponges. It's just very, very pretty. So looking forward to a lovely looking dive today. Looks like good nudibranch habitat.
We're going to come and stop there for the small samples.
Yeah, so we're just going to stop here and take our Niskin water samples. Oh, we'll have some nice whips to look at. For those of you who aren't familiar with Niskin water bottles, essentially they're sort of open bottles, so when we stop they have water from that area in them, and when we press this little trigger it basically sends a little plug shooting up to close them off, and that's what happens. And then we have our water. And so when we scroll up, we're looking to see if the little plug thing is moved position, snapped upwards.
Okay, 1, 2, and 3.
Oh, there you go. You can see the two plugs in the bottom there. Great. Thanks, Russ. These beautiful sea whips in front of us. Jeremy thinks they'd be much nicer looking if they were black corals, and I think they'd be better again if they were hosting mollusks on them. So we're never happy.
3. Right. We'll continue on.
Thank you very much. So someone has asked about where they can see the results of the eDNA work. So I think typically papers that result from a cruise get listed on the expedition website. So if you go to the Schmidt Ocean website, look up the expedition or cruise that you're interested in, and then look at papers that have come out of that. But you probably have to wait a little while. It does take a long time to carry out the work and then have it published. So quite long timelines, sometimes years. So, but that's the place you can go to easily see what work has resulted.
Control bridge.
Go ahead.
Hey Russ, are you gonna, am I stopping at the waypoint or do you want to keep the
I will be kicking you forward and just keep on this track. So I'm just going to move you now.
Okay, perfect, thanks.
Let's do a little, like 50 meters at a time.
Yeah, I'll just keep following you.
Thanks a lot.
And just as a little insight for those of you who aren't familiar with the scientific publishing process and the reasons why it can take a long time, well, firstly, obviously, actually doing the work can take a long time because our reputations are kind of ride on the quality of our work. We're really careful and we check and we double check and we test and we push and we pull until we're really totally happy with it. But then when we have a manuscript sort of analysing and figuring out what the results mean, we submit that to a scientific journal. And then that editorial board then sends that paper out to other scientists to review it. And so they may take some months to go through it all, understand it all, add their comments and insights and criticisms, hopefully constructive. And then it goes back to the authors again. And so then the scientists might modify, they might reanalyse, they might clarify some points, and then it goes back to the editorial board again, and then they might request further changes, or they might accept it for publication, or they might reject it, and then you start all over again. And so even once the science is finished, the publishing process can take a very long time as well. And so by the time then it actually, even when it's accepted, then it can take a while to come out in that journal. So very long, sort of slow timelines for a lot of that work, which, you know, I know it's frustrating for many people, but it is a good thing overall. There's plenty of time to catch mistakes or to, you know, add further insight. So it's a painful process, but it's evolved to be that way because it works relatively well. papers when we're looking at the systematics or the taxonomic affinities of different related species. Sometimes we wait because we really want to get the full, not the complete set, but we need to have very specific samples to make sense of the whole work. So we get very excited when we find things from type localities because they allow us to clarify taxonomic questions and systematic questions. So sometimes the papers are almost complete, but we wait a couple of years to try and obtain the significant samples that'll help us really wrap that bit of work up. So we all have these lingering projects, I guess, waiting for the right samples to finish it off. It's hard to strike a balance between getting work done that's sort of incomplete and not waiting too long but not going too fast and having something that isn't that meaningful.
So I've got cruises holding right on that track.
Yeah.
So we just toggle your elevation and it's keeping a good speed.
Sounds good.
And he's just creeping back all the time. So that was looking very nice. So far so good. I've just got the cruise on it, so you may have to adjust the speed. Looks really fast and the current's going to have a little bit of change.
Yeah, he's thinking the current's going to flip around here.
Yeah. Try and keep it at point one.
Morning.
I've got one on the desk, not where
Cheers.
Is it? That's where it stays on the desk.
fish here. Just a beautiful landscape this morning. Full of colour, full of fish, full of lots of marine life. It's interesting there are not so many crinoids perched on the fans here. Some other depths they're absolutely covering. Different, they definitely run on and off quite quickly, patchy.
Oh, lionfish.
Oh my goodness. Yeah. Is that the first one we've seen so far?
Yeah, it is actually.
Where? Where? Okay, maybe.
Control room deck. Comms check.
Yeah, you got it. You got it.
Yeah. I'll be taking over a bit. Ben's going to jump in and give me a hand.
It's true. Weirdly.
They all have at least one.
It's a big reliant fish
That looks
there.
like a Pegasus. Can we put a waypoint there, please?
Yep. You get that one?
That's the same lionfish or a different one?
Probably different one, I think. Anything they swim that fast.
See if it's still there when we come back.
Yeah.
That's a Pegasus, by the way. You got that way? Yeah.
So that will take up about 4 minutes.
What's that?
Oh, look at all those corals!
Oh,
Alright, I think we'll just waypoint that as coral party, please. Another coral party.
Oh my gosh.
Another
Yeah. Yes, thank you. Wow. I could sit in that spot for a long time.
So if you just go to the bottom of the screen, it comes out. There you go. And then it's started on the bottom left there. Yeah. What was before coral party? Chiaceras.
Coral party.
Pachyceras. Like Pachy. Yeah.
We have lively. Indeed.
Lost
Oh yeah, now I can see.
She's 5 meters up.
Yeah, that's fine.
Nice and scooting down nicely.
Cool. Thanks.
Or is it another one of those goldfishes?
I don't know, it could be.
Tail looks a bit weird.
Yeah. But we never saw that last time.
I think that was 54.
Very busy scene.
So yeah, now we must be about to show us.
Looks like Milo country.
So after breakfast, I like stuff.
It's breakfast dessert. Hey Ben? Ben's bringing it back. Oh, haven't eaten breakfast yet.
Your ROV drivers are bath right now.
No, it's just Ben.
I'm get that style
They're both stashed somewhere.
No, we're on things.
Karen won from I can tell.
Karen,
Oh no, not at this time of the morning. It's far too early for chocolate.
No bomb.
Save it for later.
It's very pretty here.
I think if we do a cruise t-shirt, it's going to have to be a pink colour. Mind-blowing. Even for those that have seen it, been kind of used to it, I guess, it's still mind-blowing.
We've got everything in the kind of Oh yeah.
Pardon me?
Yeah, good point.
Okay, I'm just gonna
Rich,
kill
how many
audio
ponds
for a
over
little
there?
bit while we have our morning meeting. Back soon.
So, just a short meeting today and we're back with you. Just positioning the ROV to have a look at this lovely coral. We try and not going to be at the right angle for me to see the polyp detail. Let's cheat and look at this piece. No, I think that's probably a Montipora. Not the right one.
No? Okay.
No, we can move on. It's not the Leptoceras that we're looking for.
Okay, I can just take off and we'll kind of meander through this.
Oh, actually, having said that, we should probably take a taxonomic reference sample. So let's actually take one and just one reasonable sized chunk.
Sure. Is that just not on the ramps?
And then maybe when you bring it a little closer in the arm, I'll just have a closer look at it. It's just at that distance, it's just a little challenging to see the details of the polyps.
Is that chunk on the back really broken?
This bit here, you mean? Don't
Hard to
know.
tell.
Very hard to tell.
Approach coming up.
Presume you've got an in-situ photo for that one at some point.
I did. Sure.
Just a snip of the left side there, or anywhere?
Wherever's the easiest to drop it off, but I suspect that, yeah, just taking a...
There's really a broken one. I don't know if that's broken.
We were trying this bit out here.
Yeah.
Yeah, we were trying to decide it may well have just be the angle it's encrusting, but you can give it a nudge, and yeah, if it is loose, then we'll take the bit that's already loose. I suspect it's just another bit growing at a different angle. Oh, yep. It's half attached.
Or it's a big piece or something attached to it. I could just grab the whole piece.
Yeah, you want to go in underneath and lift the whole bit up, that would be fine. It might turn into a biobox sample, but that's all good. It'll keep the coral in the best condition. I was pretty certain after seeing that bit close up that it is a Montipora, but that's cool. Can I just have a close look at it before it goes in the bio box? But yeah, do you want to get it over the tray before, just in case it decides to try for freedom?
Tries for freedom.
Cool.
Frozen.
Oops.
Put shit on here.
Can we focus that, or is it too close now? Oh yeah, that's looking good. Yeah, it looks like a Montipora, so that's cool, I think. It's very hard to tell. When these things have really tiny polyps and you can't really see the detail because it's covered with tissue, they can be a bit challenging sometimes. But yep, that's great. Which bio box? Do you want to put that in the small bio box?
Yeah, maybe 3 for that.
So,
Anything else you see here?
Let's have a little look around. There's a few more over there, but
It's the bio box back left.
sponge in the background there, trying to look like a coral. Okay, that's all good, thank you.
Okay.
Nice things.
So what I'll do is I'll probably just pop up. I'm gonna try to stretch out a little bit just because he's gonna switch headings.
Oh yeah,
So while he's doing that, we'll let him just kind of come down and around,
yep.
kind of blast up to see some cool fish up there and some life. So if you see anything, let us know.
Okay, cool.
Just stretch out.
Mind having a look at that one.
It's hard to get in there with these sponges.
So many sponges in the way, isn't it?
Yeah.
If it's really difficult, I can live without.
Just try to slide in there and see what we do.
Or if you get an angle so I can try and just get a closer look, and if it's not the target species, then we can move on.
Just look and see that belly.
Hey Russ. Yeah, control.
Yeah.
Hey Russ, can you pick up 5 meters, please?
Yeah, you're still leading off pretty good, you know.
Let's get a big belly here, Russ.
There's not much between me and the doghead. So I'll bring you up a few.
Yeah. I can stretch out.
Maybe want to string the ship out further.
Yeah, let's bring him a little bit further. So he's just making his heading change, just kind of right between two operations.
It's going to be further south. Further slow. I got this set at 85 meters. Okay, go for it, Ken. I'll just hold it.
Thank you.
Yeah, well, he's turning and just punching a bit ahead. That's the doghead's leading. So
Okay.
45 degrees to your starboard side of the vessel.
Roger that. Thank you.
I'll keep an eye on it. Just
Ask him if he's going to turn more to port.
Yep, that is, of course, one of the ones I'd like to sample.
It's fine. I'll get it. So 180. Yeah.
Okay, I wonder if we pause him there and then he can rotate instead of trying to cover this.
Bridge control.
control, bridge.
Hey, Luke, can you pause your turn for the moment?
Sure. Yeah, I'll pause everything.
Or sorry, Luke, are you happy to pause there and continue your turn?
I can do that. Yep.
Because you're going to come 180 heading?
180-ish, yeah.
Yeah, okay.
Maybe not 180 to begin with, but
Sure.
switching over.
What I'll do is I'll put the waypoint right about there. Just kick ahead a little bit and then
Yeah.
continue your turn.
That's perfect.
Cheers, thanks.
Thanks.
So, can you reach out? Let's see. Yeah, it's the, oh yeah, oh, you reach out. It's that one right there.
Yeah, right in the middle.
Just a snip of it?
No, a large chunk. If we're lucky, it might just ping off because it's probably about that big, so that'd be nice. Oh, and that's probably another one down there too.
Can you swing to the left a little bit? I don't know if there's the box. I think you can. I don't see anything over the left.
Just sit on this little rock here. Don't worry. It's over and it drops off.
Let me try.
It's this one here.
Just try some shots. Yeah, this one. Are you ready, Karen?
Did we get an in situ photo, guys?
Yeah, we did.
Thank you. Yes, we're good to go.
And you want a big.
Yeah, maybe if you just try going underneath it and just nudging it, and if we're lucky, it'll just ping off. But most likely, it'll probably just crumble. They only ever ping off when we want a small piece, and they only ever crumble when we want a big piece.
Can you zoom in? See if that base is thicker in the back. That's close together. Push from the back, it should pop out. Right, zoom out a bit, please. Just hold this man out. Try and pop it out. Karen, can you zoom in a little bit?
Oh, you've got it, Adam? Thanks.
Just pivoting on my last bumper.
It's very hard, Karen.
Yeah, it's most likely to be attached where that central polyp is there.
So I was trying to take off.
Yeah,
Yeah, it doesn't have much leverage underneath it because there's not much access.
yeah. No, we just take it as multiple smaller pieces.
Yeah, I'm gonna have to do smaller pieces, Karen.
That's fine. Yep. They always land at the worst possible angle, don't they? pretty well attached, isn't it?
I think just takes it. Yeah.
ROV bridge.
Yeah, go ahead, Luke.
Yeah, I'm just gonna keep this heading for a while.
Okay.
And see how she reacts. 215 heading.
Sounds good. I just bumped you a little bit to your port.
Okay.
Yeah, you can, if you want to sit there too.
Slide on over.
Sure. And if you keep rotating, just let us know. We're in a good position then, I think.
Okay, great.
Cheers.
Thanks.
Okay, off.
Go ahead, bump up again. Just make sure I push down a little bit. Try to get you a little closer. How's that sponge on the right hand side? It's good.
There.
Stand by. Get frozen there. Just on the one corner. So.
Stable.
Yeah. Yeah. Just the way it perched here.
Beautiful. think that's probably enough of that one. Thanks. Now there's one that's actually just down in front of us that I'll have a quick look at before we move on.
Approach out a bit.
So it's that one there I want to have a look at. Oh, there's a little Pachyceras there too. Yep. Is that in an incredibly difficult spot to sample?
We've got Ben. Ben, grab it.
Okay, I'm just going to look at this other little guy first, which looks like a little baby Pachyceras.
This one's
Oh, pretty. That one's probably a bit small. There's my lasers. Oops. Where's the other laser gone? There. Now that one's too small. But yeah, this bigger one, if we could sample that, I think we'll take the philosophy of if it breaks, we'll just take a small piece, and if we can get, you know, if a lot of it pops up, then we'll take a big piece. How's that? Oh yeah, can we just take the in situ shot before we put the porch back out?
I might be able to come down with the ramps on with better access underneath there.
Oops, just a smidge too far. How's that, Declan? Good? Oops, I can just zoom in a wee bit more on that one. Just get a bit of that polyp detail.
Just give me some zoom.
Get that too?
Yeah.
Thank you. All right, good to go. Thanks, gentlemen. Oh yeah. I think it's going to be our challenge all day. Is trying to squeeze between the sponges.
That's right. I suppose that's better too much than too little. Okay, that's the okay, then. Just porch a little bit. Team porch. Is there?
Oh, that's a nice big sample then. Fantastic. We could do that in the bio box. Thank you.
Photoshop. Karen,
Sorry?
not a shot.
No, it's fine, thanks.
Okay, about box 3 for
I reckon you can put it in that same one we put the other one in, to be
far
honest.
left.
Yeah.
Put it in the same bio box.
Yep. Beautiful. Can we look around just over there? No shortage of coral. Is this where we marked the coral party?
Yes.
Yeah, it looked familiar.
I'm trying to sit down on this ledge and give you a look around here.
Okay, thanks.
Okay.
What?
Actually, let's just swing around to the other side. Better location.
2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. At least 15 colonies there. Not all the same species. It looks like there's at least 3 or 4 species there, which is interesting. But there's actually probably more than 4 species. I think there's probably at least 6 different species in here. You're comfortable?
Yep.
All right, have a good look around here. This is beautiful. Kinophilia. Oops, wrong way. Can we focus that?
Just try to get a little better there.
It's funny how sometimes you can just see the detail so much and then other times it just won't resolve.
You zoomed all the way in?
Zoom in? Yeah, it is now.
Okay. Yeah, you can pay out about 5, Russ. Thanks, yeah. And try coming back in.
Karen,
It's almost like
any
it's...
idea what this dark on the left is?
Sorry, hang on, Amy. We're just trying to get focus on this. Oh, it's a bit better, is it? I suspect part of the challenge is that it's so variable.
Copy that, thank you. Yeah, something to some.
Okay. Thank you. I can see enough. So yeah, it is a Leptocerus, but it may well be a different species to the one that we've been sampling. Right? Which one were you asking about, Amy? Sorry?
Do you know what this is?
Oh, that's one of the echinophilia type things.
Okay, 'cause, yeah, we were sampling, we've got a few samples of it actually for ecophysiology, but it was in the quite the juvenile stage. It was kind of only like this bit around the edge. So yeah, I was just curious because we haven't seen a big one in close up. Yeah.
There's actually quite a bit of it. There was quite a lot of it at the big mega site we did last as well.
Yeah, I
It
just
kind
kind
of
of about
happened.
two or three samples.
Yeah. Do you want them from here as well?
Yeah.
If it's easy, if we've got time, but it's not a big deal.
Right. We need the replication, and we've got. If you're starting to get replication at one location, then it's pretty sensible to get it at a second.
Yeah, we could do 2 or 3. Yeah.
Okay, I'll just keep looking around first.
Preference is definitely the Tachyceras and the Leptocerus.
I'm just gonna try to put you back in auto there. There you go.
Ah, that's better. Thank you. focuses this. Oh, are you thinking more of the more thick line things rather than thick line?
I'm not sure because it didn't like the edges of that.
Because that's the
had
same as
dark
that
central-
freaky blue green thing.
Okay. So that one
We took samples of, there was just small colonies.
Yeah,
And they had
there's a Lepiceros that's sort of more heavily lined. It was somewhere halfway between.
Yeah, it was like that. With those lines around the edges and the darker bit in the middle, like really distinct lines coming out. And the skeleton was really fragile. It kind of just was breaking into pieces and still stuck together.
Yeah, we're just kind of sitting on this bridge right
It just, yeah, it really just looked like that. Sort of, like the,
Does it look, is that more like it?
yeah,
That one
it
there?
looked like that one.
And that's still, that's considered for whichever genus
It
it
looks
is.
like a small version of it, yeah. I had a big question mark in my description and it just said, yeah, distinct longitudinal lines with dark
Maybe do better too if you just keep it as tight as usual. Forget the top floats. Oh yeah, the top floats are 8 meters, 5 meters closer to them. That's red beans and rice for lunch. I like some red beans and rice.
there's your
Didn't miss me.
Oh, come on, focus, focus, focus. There we go.
I think they're like a meridian of the day. It's like a disaster.
That's a great colour.
Whoa, that's interesting.
That's our fancy colour to kind of fill in.
Too morbid, but we find out it was short jumping styles. So with bad guys, it was short, bunch of jumping styles. Okay. Yeah, they're gonna put that. Hey, weird news. Coca-Cola's average sales have dropped 11%. These chips.
Oops.
Ben
Stop,
Jerry's
stop,
on the
stop,
brink
stop.
of bankruptcy.
That's out of reach, isn't it, up there? Just out of interest.
What's that?
Is that out of reach up there? Oh, you guys have changed when I it's not critical. There's a bunch here. I'm just sort of deciding which ones of this selection I can sample from this location.
Let me see.
It's probably a bit far away, isn't it?
To the top or the
This one here.
yeah.
Too far away? That's okay. There's a bunch of others, but I was just I was just making my shopping list mentally in my head.
Try to work our way up.
It's okay. I don't think there's a shortage of other ones. Like I said, I was just mentally making the shopping list.
It's like a child.
Wow. 65.
so.
We're going to take our auto heading up, try to keep our temperature cooler. So it might move a little bit just to let you know, but then I'll get right back down if it does.
Oh, okay, no worries.
Alright.
Yeah.
Oh, nice.
Turret
Put a
deck,
name on that
just
one,
to give
eh?
you a bit of information. The locals look quite nice, straight aft.
Try and put a name on that one.
It's only leading
It's a
maybe
challenge.
30 degrees to vertical.
See the structure very well.
Talk about it.
Did we just get Irish in here?
We did a little bit. You sound like a little bit.
Maybe a terrifying one.
Is there a sediment too? Can we get closer?
See if I can find a better shot for you.
It almost looks goniastrial like those that kind of hexagonal loopy shape.
Yeah, no, goniastria doesn't do those frilly things as a rule. Yeah, let's have a look, see if we can get this piece of it. This is why everyone hates spatters. Or blue line, it's more to the point.
I can't even tell the shape and definition of the lines. So,
It's a challenging one. I thought it might have been the Lactastria, but it might be a bit big for that. It's a bit hard to scale it. Right, shopping list. Okay. There is a huge diversity there of different corals. Let's start with this little easy guy at the front, hey? This guy here. Can we collect that if we're happy? If we can get that whole bit, that would be nice. I'm hoping it does the pop-off thing. And if it doesn't, we'll just take a small tissue sample. Oh yeah, the whole photo thing. Sorry. I keep forgetting to take the in situ shot. I'm just excited by all the corals. Oops, and then upper wing. And of course that soft coral's just right in the middle, so we can't see it properly. Alright, does that work?
That works very well.
Thank you. Alright, now we can go. Thanks, Cody.
This looks strong.
Oh, there it goes. Thank you.
I would have an open one in the front.
There is an open one, yeah. Number 31, I think.
Fits, if not,
Yeah.
stand by. I want to do this. Yeah. Did we grab them? I'll slide one. Alright. Unfortunately,
Looks like the perfect quiver size. Hopefully.
Alright. How's that?
Hey, Connie, I'm just going to hand over J-Rod here, and off comes.
Roger. You still pull it up. Yes, sir. Well.
Oh, masterful.
Cool. Now that's not annoying. What last time? What was it? That didn't really help a lot. Sorry. The intention was there. We've been collecting a lot more in this spot, right?
A few more, yeah. Do you want to
No, you're fine.
go for a swim or
No,
Oh, you're okay?
I haven't get another lid off before I
Yeah.
pushed it. You're not gonna mess that anymore? No. No? What do you? Alright, we're gonna collect a few more here, so if you want to just prepare that, go right ahead. Okay. Happy 20th, everybody.
About 3 more?
Yeah, I think that's probably a good number.
It's 18 broken. That's, yeah, it's just. How that happen? The physics behind it is bad.
Thank you. Let's sample a bit of that one. Just get my in situ photo. Can you do the in situ photo for that one, please?
Yep.
And I'll just zoom in for some good polyp detail. Good.
Yep, all good.
Alrighty. Same principle. If a big piece comes off, that's great. And if it doesn't, we'll take a little piece.
You want that little nub on top?
When you say the little nub on top, which one do you mean? That one or that one?
That one, the second one.
This one?
Yeah. That right there.
You could give it a go. It's either gonna be the toughest part of the colony or come off really easy, you know.
We'll see, right?
Let's find out. Probably at a good angle, at nothing else. Catch may be that it might be encrusting up over a piece of harder rock.
Son? Watch out. Alright. So that looks like it's a piece of rock.
It does look like it's encrusting over a piece of rock.
Piece.
Yeah, this is my situation. Watch out much more. It's gonna destroy that. She wants.
Watch out.
Yeah. Alright, you can do it.
Go ahead.
Sorry.
Just to... There.
Woohoo!
Nice.
Would you like another piece or is that good enough?
If we can get another couple of that size piece for this one, that would be nice. Can you grab that bit? Or is it at the wrong angle?
I might be able to.
If it's too challenging, we can just come and get a bit down here. It does look like it's at just the wrong angle. And with a rock in the way. Oh. Beautiful.
Get up that outermost screw on that side. Alright. Which I will look for it in a second. Same spot?
Thank you.
Alright.
Close it.
One more that
One more.
size, if that's okay.
Okay.
Then we can use it for the ecophysiology, the taxonomy, and the reproduction. So that's great, and the population genetics.
Piece of, is that piece too small right there?
I'd grab that piece if you
The
can.
one right on top?
Yeah, yep,
Where the rock almost kind of.
yeah. Waste not, want not.
That's it, brother. These mornings like this, I wish I drink coffee. It's a crack in the middle, so just be careful. Yeah. Yeah. See what that does for me. It might, if I pour around, it might getting slow, so we'll start her out. Come on, push something. What is anything? Alright, that's fine though, because it'll keep this thing spinning too much if there's anything over there. Alright, let's try for it. Wow. Good on you. I didn't touch on these things. It's impressive. Nothing. Can't do it. It's gonna swing inboard and try to get that left edge. That's on the wall. Just thinking about that. It's pretty up here, right here. It's a decent piece of drift. Yeah, well, you're.
Right, man.
Yep.
That's what I'm hitting around.
It is a rock shape with the top of the floor. There you go.
Good.
Hold up.
It's off my right.
Worked out.
Oh, that's two things.
That's a bit more than quiver sized. Oh, it'll probably go in a quiver.
Must be drawn on a sponge. That stuff.
Yeah, it looks like it, yep.
No.
Everything about this coral is just the wrong angle for everything, isn't it? You're doing a great job, Cody.
Right there. Alright, you're over it.
Beautiful. Thank you.
Close it? Or you want me to grab
Yeah,
some?
no, let's close that one. I think we've done it.
Fair enough this morning, or?
Next. You happy with there still?
Yeah, what it is.
I'd like a piece of this one. Just
Alright.
one sort of reasonable sized chunk.
Everyone gets a picture, right?
We do want to get some pictures, you are correct. Could you take an in situ photo of that one, please? And I shall just try and zoom in and get a little bit more polyp detail.
Is this also a Leptoceros?
It is a Leptoceros, but I think it's probably different species, which is why I'm taking a sample for taxonomic information. Now, where did we focus it on before? That's right, we got it to focus a bit more over there, didn't we? Oh, stop moving around. There. Okay. You can get a bit more detail on that. So cool. All right. Yeah, just somewhere on this edge, just take a
Alright.
whatever comes off.
Anybody watching it?
No.
Yeah.
No, we don't want that big a piece. Would it work if you came in and
This right
bashed
there.
it down there?
You want like this?
Yeah.
Maybe I can probably stay down. Stay down. Okay.
Ah, yep, that'll do. Thank you.
Now I'll go back for. You want that piece, right?
Yeah, I want the bigger piece. Will that bigger piece go in a quiver, do you reckon?
Yeah.
Good.
Let me throw this little bits in there.
That's beautiful.
Watch out.
Fortune out. Settle down.
Really?
Really? Since you're gonna brisket, you gotta get the edges off. That
Really? One of those mornings. Temperature.
65 degrees.
I just have one this side.
brittle those ones bring my back and they're fairly tough in the lab?
Sorry?
How brittle are those whenever we bring them my back? Are they fairly tough in the lab? Or are they
Yeah, it's actually much harder to get a piece off them than you think sometimes.
when it's
Oh,
10?
and a bonus soft coral. Sponge, actually. Bonus sponge.
That's a big inch, two big inches. See it back there?
What?
The Moorish idols.
Real. So wider than I thought. It's spinning. Normally.
Oh, that's a good way of getting in. 2 small pieces. Perfect. Good thinking. You'd planned it that way, hadn't you?
Yeah, it's over. And it says that the ice is
Yeah, well done. I think we might just sort of move a little bit around to there. There's a few more. Just a little bit around to sort of in front of this spot around here. I think there's a few more Leptoceras hiding in that direction.
alright.
But we certainly can't see them properly from here.
Which ones are you interested in?
That one, possibly that one,
Okay.
and I think there might be another one sort of, and I want to have a closer look at this one. And I had a closer look at that one, so somewhere around there. Oh yeah, there's a suite of them there. If we could. Oh.
Which ones?
This one, those ones, and looking at those
Okay.
generally. So
See, I'll just come around like that. How's that angle for you?
that's beautiful.
The only thing I don't like is that guy right there. So, Looks rough, right?
That looks pretty good.
Yeah, well, I'm just saying that. How's that? What?
A little bit more forward.
Forward? Yeah.
That's fine.
That needs to go for the process.
There's at least 4 things I can sample here, so I'm happy.
For a meeting, like that.
Oh, okay. Alrighty. Yeah, I'll bring my back. Yeah, thanks. So we're going to want to sample this guy here in the front there, which is a Pachyceras. Yeah, the stripy one, if you like. Terrible place for a photograph of it.
Sorry.
No, it's all these fish in the way. Okay. Ugh. Can we focus that a wee bit better or not? Or is it just, it's just going to be difficult, isn't it, with all that stuff in the foreground?
Yeah, a little bit.
Yeah, you're right. You definitely about 5 to 10 meters.
So we've got an in situ grab. If we could get either a quite a large chunk or 3 palm-sized pieces again of this one.
Okay. Ready to get it?
All good.
Yep,
All right, going for it.
it's a go.
Let's see what happens.
Thank you, sir. That doesn't smell right. Sit for you. We're gonna have to take.
Going down.
Hey, welcome back to Sea Love World.
Yeah.
Sea Love. We're on Falkor here.
There.
We're at the north. Ashmore.
Just ignore just in the front. Head like this. Do that.
Today we are collecting a Pachyceras icon.
Stuff.
There's a rock there.
Hey, we're in this spot.
whole thing comes up. YOLO?
Break it into pieces.
YOLO. We'll try not to take that.
I don't think that angle's
How
gonna
big
happen.
is the whole thing? Oh, it's really crumbly.
It's about this big.
It's extremely crumbling. Crumbling.
Like an apple.
Like really, really.
There you go.
Oh,
That's
there
good
we
bit.
go.
There's another bit.
There we go.
There's 2 bits.
Hey there.
That is a crumbly bit.
Now we've just got to LeBron that right to the river.
That's a big enough piece, right?
For one.
That's pretty big.
Cool.
That's kind of gotta fit over. The edge there. The big edge.
It's very crumbly, so it might slide.
Alright, fortune out for you, buddy.
Plus, I got tons of crumbled big pieces in my What source is it?
Whoa. Oh yeah.
That's perfect, right?
What?
It's 2 pieces. Explore.
Look at that makes me want to have a tomahawk steak. Here you go. Go to right, just to touch it. There you go. Just a little bit more. There you go. That's it. That's it.
Is that in? You want more?
If it's possible to grab another.
Okay.
Same size or
Just that one.
that one's about that big.
Yeah, probably need.
Another one of those?
Yeah.
Another one of those, if not bigger.
Another one of those is fine.
Beautiful.
I'm really bad at judging the size from the screen, so
It's okay. We'll grab it.
pretty much for the ecophysiology study,
Okay.
the minimum is like 3 pieces palm size.
3 pieces boxes.
If it's less than that, it's not too much I can do with it.
Okay.
So I'll let you guys be the judge of that.
Same ones like a fly.
What's up with this? Even, how even is it's down to me. Because I could encourage gravity.
Fish, move.
Like that's the closest thing. Trying to work here. Let's go.
You can get that back edge.
Right here.
Yeah.
I think I'm under.
Yeah, you are. Not a whole lot, but yeah. You're about half your. It rises up behind it. You know what I'm saying?
God, yes.
There you go.
Good job. Nice.
That's a nice size one. That's about the same size. Just shape different.
Amazing.
Go ahead and bring that little porch bag out.
Alright. That's good. I'll build this bench. Or take a shower. Take the spread of the brown.
So you go, man.
Is that good?
Thank you.
That's great, thank you.
Welcome.
What mix is that?
That's good. 20.
Number 20. 420. It is. What a great. I
3 weeks.
Actually, go kind of nuts like that.
Yeah. Still on my break tomorrow. Look at those fish behind us. Oh, they're gone. They're back. Porching out just a little more. I'm not a big a fan of those ones or the older style, the one that 19 is. It's not so big of a
Yeah. Okay, next.
Alright, so we're going to take this opportunity to look at what we want. Anything from you two you want us to look at while we're waiting?
Did we already take from that one on
We
the
did
right?
not.
We did not.
The only,
Okay.
the only thing we've taken so far from this location is what we just did. So
Okay, could we have a close look at which of these things?
Jump in the sea.
a lot of pressure.
This bit here.
Introduce yourself first.
Yeah. Tell the people at home who you are, where you're from.
Hi, Amy.
Hi everyone, so this is Amy speaking with you at the moment. Oh, as
You keep
Karen's
going,
just
you're doing a
walked back
great
in the room.
So you got the Pachyceras?
Yeah, so we've got the
do you wanna
Pachyceras.
have a look at a couple of those
That's
Leptoceras.
exactly where I was just pointing. Fantastic. So I'll be carrying out the ecophysiology study on the hard corals. So we're just going to look at this section on the right here, please, of these colonies. Oh. I don't know how to move this.
Right's right, up is down, down is up. Right is spin right is
Doing a great job.
That's really smooth, Amy. Look at that.
This is smooth.
It's like you've been doing this for years.
Oh, I'm a natural.
which one would you like?
I think we can zoom in on this one, please.
You're the one.
It's really good zooming.
Smooth.
It's better than carrot, right?
I'll go ahead and focus that, see if that helps you out. There you go.
Oh, yeah.
Get out of there, fish.
Yeah, okay, so that's the Leptoceras. We might take just a small sample for PopGen.
Okay, so pop in sample.
We'll see how big the piece breaks off when we touch it.
Alright, we don't have anything open.
No.
Alright, so I'll
Is that fine for you in situating? Happy with that?
Yeah.
Okay.
Thank you, Declan.
Can we just do 20?
we just did 20.
21 available?
Is 21 available?
Hey, Declan, is 21 available?
21 is available.
Sweet.
Alright.
So just to clarify, we have 24, 23, 22, and 20 taken up, right?
And 31.
And 31. Okay, cool. Thank you, sir.
No problem.
Give me a reshot.
Which one? This one over here?
Yeah.
So how big roughly is that colony? It's about
About that big.
This one?
15? Yeah.
Yeah.
So we'll probably just take a little bit off that.
A little chunk?
Alright.
Yeah, unless the whole thing pops off, we'll grab it, but
Okay.
should be okay just for the PopGen.
Fortune out. I did see that.
Fish.
Fish getting a little aggressive with us.
It's a territory.
I think that the more we mess around, the more food pops up for them and they get all excited.
Which one?
Oh, hey, buddy.
You happy?
Alright.
It's Johnny Graham.
Because I've just read one of the comments, someone has to watch this for biology class, which is interesting.
Fish.
So at the moment, we're looking at some colonies of hard corals. There's a lot of sponges, there's some soft corals like the white one on the right of the screen at the moment. And at the moment we're collecting this main colony of corals in the middle of the screen, which is a species of Leptoceras, which is a common one that we've been seeing across all of our sites, and one of our main specimens that we have been collecting as well for population genetic studies and also ecophysiology study.
Sorry, you're getting the whole thing.
That's completely fine by me.
That is going to be.
We can use it, nothing will be wasted.
Alright, so 421 will still be. That's going to be. File box which is.
Yes, we'll pop it in a bio box. Will it fit in bio box G, right?
It should probably fit in that right there.
Yeah, that sounds good.
And do that. So we're going to put it in the right side and the left side. Okay, middle port.
Little port.
Waiting for you.
It's like the port.
It's starboard port.
Is it like F port?
No, it's not aft. Well, they're all aft, but it's starboard port.
F port?
No, not aft forward. I don't know even what you're talking about.
Forward aft?
Just a question to all the viewers out of sheer curiosity. How long have you all been tuning in for the dives? I'm interested to know how many hours everybody is sitting down for and watching.
800s.
Had 4 hours, 6 hours.
12?
Okay. The question is, when you go home, are you gonna start watching this?
That's good. That's great.
No, when you go home, are you gonna start watching?
Sorry?
You go home, are you gonna start watching?
Maybe.
I think that's.
Not as long as these guys. So we've had some people say they're staying on for several hours and the whole dive, which is really impressive. I commend everybody's dedication to the dive.
When we got our next cruise, we'll be doing 24-hour out, so we'll see how dedicated they are. Alright, now what?
Okay. Did you already take from this one?
Now we took.
Okay, so we'll just have a closer look at this colony on the left. Because the first one we took from this area was a Pachyceras.
So we'll shoot this Chris purple and yellow.
That's more blue. That's more of a Michigan fish.
That's definitely
That color yellow is actually more Delaware. To be totally honest.
Oh, looks a nice spotted cod type fish down the bottom there.
That would be a Florida colored fish.
Can we get any closer to those polyps to see more detail on there?
That's all I got. I can move if you need more.
Yeah, cool. So we will probably sample this. Can you get an in situ of that one?
Sure can.
Declan, thank you.
Seventh colony of the day.
Okay, so I'm hoping that one breaks away quite easily, which would be great for ecophysiology and popgen. Otherwise, happy for a small piece as well.
Might have to go for that. We'll find out.
Is there, where's it at, Chris?
It's right there.
Oh, yeah, you're gonna move that. Is there any ones that you want closer while
Before
we're
we
right
move.
here, before we move?
Okay, so is that a fair while away?
Yeah.
Yeah.
Okay, no worries. We'll zoom out and see what's there.
That's not-
Just out of curiosity, can we have a look at these ones? Just for some photos
These dark
might be
ones?
nice. Yeah.
Sure.
Great.
because these are really abundant in this area, which
Like
is
an
cool to
overview
see.
photo like that, and then get a little closer.
Yeah, it'd be great.
Alright, you got a picture there?
Have a nice close-up look, perhaps in the area where there's not too much sediment piled onto it. Ooh. Screen grab for me. Awesome.
Yep. Good fish. Very nice.
Goes can
Someone would love
How do you zoom out?
Spin it on.
Oh, I see.
Just do it nice and just don't let it go. Just do it nice and slow. And then slowly
Alright.
keeps it jumping too hard. Just remember if you want to go up, pull back. If you want to go down, push forward.
So much life in this area. It's awesome. So I think because we're not going to be sampling these corals here, we can probably move about now.
I already got one right in the middle.
Yeah, if that's easy to grab. If it's a bit hard to grab, that's fine. There's quite a lot of them in the area, so.
See, this one right here. Correct.
Yes.
Whoa, that was unexpected. Alright. So,
That's gonna be a little.
so we will take off and
Yep.
stay in this
So
general area and see what you like.
we're not going to get the Leptoceras colony that I was looking at because we don't want to be breaking any of these nice big colonies down there. So we're going to find a different location with another colony that's more easily accessible.
Do me a favor. Bring your arm in for me.
Okay. Is this easy to grab, this one? Just sit here.
Your check coding. Where? Right in the middle.
the pancake one.
We'll just zoom in and make sure. Yep, that's one of the common ones that we've been collecting. So if we can get an in situ shot of that
Yeah.
deck then. Could I see the green dots? Just there's a lot of fish in the way.
Yeah, just Alright, I'll back out with the laser.
Ah, looks like there's a little crustacean underneath it as well.
There you go. So it's about 10 centimeters wide.
Yeah, so we really only need a small portion of that.
Okay.
That will just be a population genetics sample.
Yep.
So will this work directly impact your schooling, or is this something you're just doing in addition to it?
Oh, okay. So my entire research project for the year is based on the data I get on the Falkor.
That's a lot of pressure on this.
Yeah. So we'll be collecting all my samples and then analysing them back in the lab and then writing up a big report and thesis and hopefully some papers to publish towards the end of the year.
If you do publish papers, my name is K.
Grab your grab.
I'll definitely do a shout out to the Falkor crew.
Appreciate it. Oh, scared little guy. I'm moving too much. Let me know. I'll oops.
To the right.
The right side.
Beautiful.
That's all I got. Do you need me to settle out a little bit more?
No, it's inside of the rock. You said you just need a little piece, right?
Yeah.
Oh,
That's pretty hard.
Oh, is it?
No?
No. Well, yeah. Normally. Going so much.
Alright, yeah, I can, if you need 2 seconds, I can check the grams up, stabilize this. There you go.
Yeah, that's perfect actually. Perfect size for population genetics.
And you got 2 of them. Pizza shapes.
Alright, portion out. Right, here we are. Let me porch out some more. You're right above it. In a good place. Happy place. There you go. Yeah.
That's perfect. Thanks, guys.
So Joel, I was wondering if you know how many species of the source.
Okay, so we've had somebody ask as to any guesses on how many Leptocerus species are down here. We're not too sure. We think we've definitely seen at least 2 or 3. But as you know, it's very hard to identify many corals down to species level, which is why we'll be taking the DNA samples, which will help us get that number of species to more accurately.
Okay.
It was probably about 3 or 4.
You want a quiver probe? Do you want a little job? We'll sit here on the look around if there's anything else you're interested in. He's gonna take a lid off and then we'll set you up to look around if there's anything else.
Sounds good.
I might not take too long.
You do whatever makes you happy, buddy.
That's not an option, I was first.
It is. It's just it involves a lot of swimming.
Long flights.
Yeah. When to go home and be home for like 4 days and a week, and then come home for like 7 days and a week again. Then we
Come out of it.
Yeah. Just have to tighten that up and get up there.
So we're currently sitting north of Ashmore Reef. The biodiversity is really high at this area and we're at 54 meters.
Yeah. very warm, so that probably contributes to that.
So one of the scientists who has studied shallow reefs at Ashmore, Zoe Richards, who's also the coral curator at the WA Museum, has given me some information and told me that there is a really high biodiversity of corals on the northeast corner. So with that information, I'm expecting to see some high biodiversity sort of at these 50, 60 metre depths, which will be our site for tomorrow actually. So seeing as the biodiversity is quite high already, that's very promising for tomorrow's dive and the day after as well. Okay, let's have a look around.
I think you're gonna be like 10 minutes looking around. Go work, grab the coffee.
Probably not.
Stay there. You're having fun. Stay there. I got so absorbed in the corals, I completely forgot I was meant to be in there. Which is why I ran out.
That is totally fine. I'm just getting the hang of the camera.
So what did you decide the other coral was, by the way?
Which one, sorry?
The Amelia liner. Did you have a good look at it while you were there? No.
We had a little look at it, but I still don't know what it is. I can't see the distinction very clearly.
Good challenge. I think I've worked out with this, so it's your challenge
Can
to go
we-
and learn it.
Focus this one. I am looking forward to looking at all the skeletal samples we've collected and doing some taxonomy work on them. Are you distinguishing between this as like species 1, species 2 of Leptoceras, or
No,
just
so
grouping
the
them?
second one that, the one that I put as split 2 was that one that was really lined, really wavy. It was
Okay,
sort of
cool.
like a halfway between. So this is just the normal one. So that's
Great. Good little pop chain sample for you.
Yeah, please.
Yeah,
Awesome.
we won't be able to get that from where we're at, so we'll have to move. So
We
if
went-
there's
okay.
anything to the right. is if
Yep,
you look-
not a
if you
problem.
look-
Okay, we'll have a look at that first.
If you look down here, let's see which direction the camera's going. Pretty much everything
Okay, sure.
beyond that thing being straight to the left, you'll be able to reach.
It's a lot of little fish.
But we can
That's
just
fine.
move over and get that in a
Custom
minute if you really want that one.
You want to zoom in on this one?
I think it looks like a sponge from this distance, but have you had a look at it? Oh, you just landed, sorry.
I haven't had a look. But it did look like it's really stuck onto that rock. Might be very hard to sample if it's a coral.
Oh no, it's another one.
Oh,
before,
yeah,
isn't
it does look like a Montipora.
And out of focus.
We can
before, yeah.
focus that one.
Oh yeah.
Cool, so we think this is a species of Montipora. So it's got those really, really small corallites. They aren't very distinct. So we've had some people that have been told to watch this for biology lesson.
Wish my biology lessons had been this good.
Me too. Oh, and look, we've got an... I'll go up slightly. It's wrong way.
All
Up,
the other
down.
up is the case maybe.
So this is a bluebell ascidian.
Were they called bluebell cities?
Yeah. So you can actually see them quite a lot around Perth
Yeah.
as well.
It's like an eyeball right there. You see that? A white eyeball.
Oh, that's
Really
kind of familiar up there, isn't it?
beautiful soft coral here as well. And we'll just take a look at this coral in the background. Oh, okay, that's
No, it's a
Leptoceras.
Leptoceras.
It's a Leptoceras, so this has got a really distinct polyps here. I think they almost look like little candies. Have a look at lots of fish. Now I know there's been a few people talking about the fish, so I thought, I was going to have a look at that emperor down there, but nope, he's gone off the screen. Not too sure what
That one's
coral this one
probably
is.
picked in at the sunset. I was actually just in this bigger one. Is it the same thing?
There. I think it might be. What's your verdict on this one? I'm not too sure what kind of
It's
coral
too hard
this
to
one
see
is.
the detail.
It
Could even be
is.
something from another multiflora. Something good like that.
Okay,
Without
so
seeing it
I think we've sampled all that we'd like to sort of in this area.
So you want to go to the left and get
Oh,
that
did you
one
want to
that
get
you're
that Leptoceras?
Yeah, so we've just got to move to get the next Leptoceras colony that we've spotted for collection.
Which one was that?
So we'll
Right.
aim for this one,
Alright,
yes.
and what about the one right there to the left as well?
And yeah,
Both of those?
either actually. This one's probably easy to
Yeah,
grab,
that
I
one'd be
think.
easier.
Yeah, we can go for that one.
Alright. Hey Chris,
Yeah.
before you go, look up to your right. Bunch of fish are eating a sponge. Just chowing down on it. Right there.
Oh, up there.
Look at it.
Oh, wow.
Oh yeah.
I pointed it out. They're eating something inside of it. It's pretty cool.
Circle of life.
And so you can see the butterfly fish in the background just eating at the sponge. So the butterfly fish actually also eat coral polyps too.
Look at their little mouths.
Can we get a couple frame grabs of that?
Truly out. Woodpeckers of the sea. I like it. I like it. Get young gangs. They're berets. Speak to the bones. That's right, just
Celebrity fish. Alright, Cody, we're gonna go up to that guy right there. Let's see.
the one with the candy on it.
How many did you manage to sample while I was?
Maybe 2 or 3. So we've got a pop gen. There was a Leptoceras that kind of popped off. Potentially 1 here while we were still over there. Yeah, it's maybe 2.
Please don't do that.
Oh, sorry. You're looking where we're going.
So have you got your minimum of 3 big ones now? 'Cause this one's got enough in it, I think.
Yeah, so
This
I
one's
think we've
got
got
enough in it.
1, 2,
3. So this we've got
3,
number.
3 Leptoceras.
So that's great. We'll get a couple.
Hoping they're all the same species. Great.
We'll get a couple for luck. But so long as we've got minimum, then we can relax.
Okay.
This one?
I thought you wanted me to do something before I said.
Oh no, that's fine. So just with regards to the butterfly fish that we saw eating the sponge, so I've had a fish ecologist from Perth just send a note in to me, Kurt Crispin. So he said that it's quite unusual to see the fish eating the sponge. They normally don't go for them because of the spicules on there, and yeah, we've just noticed that we haven't really seen that much. So that behaviour I think is a bit unusual. So if there are any other fish ecologists watching on, your opinion on that would be great. So we've just had a coral about the population genetics study, so that is more Karen's domain. Did you want to shed some light on that, Karen?
Yeah, so Kai, we will be using SNPs for this particular study.
Okay, so we really only need a small sample from that colony for population genetics.
Alright. Yeah, fish on there. Very good. Over on. Weather. See you later. I don't want
We haven't moved past the coral party waypoint yet, have we?
We've moved around.
Yeah,
Don't know how far
because
from
there's
it
a
we've
second
gone.
coral party waypoint.
Should we head
further
there
away.
after we grab this specimen?
Oh, we can still keep looking.
Yeah, I think this is one of the
We've
last.
probably done a full circle around it now.
There are some colonies inwards, but we'd have to move the ROV, and we don't want to be breaking any of those beautiful big colonies.
That's the challenge when there's so much diversity here. And
That's
so many
it.
things. It's hard to find a good ROV sitting spot. Don't want to do any damage.
Yeah, so Karen is definitely best to say anything about the population genetics study. However, if anybody has any questions on the ecophysiology study, I'm more than happy to answer those questions.
Have you photos? You ready
In
to photo?
situ shot?
Yes.
green light to grab.
How do we describe this thing?
So that would just be Leptoceras, and that will be a population genetic sample for when we do the sample. Oh, it is. It looks very flat.
Yeah, it looks like it'd be hard on a sample.
Yeah, I think so.
There's a big Pachyceros that I tagged on the way across.
Beautiful.
We've got one. Did I get one already?
Yes,
Oh,
we
I thought
do.
there was one that was.
There is, yeah.
You got some back off there. You can grab that.
Like a bit of shit.
Rotated right.
Oh, that was one I think.
Yeah,
I did
yeah.
Just when I ran out.
Come up.
Oh, probably left you.
I'm up with the opportunity to move back.
Gotcha.
Yeah, nice and clear.
It's number 20.
Hold up for you. Sorry, I'm being considerate.
Another typo. What's that? Another typo. I just grab it for me.
And that piece at the bottom of that, let's see that off. Alright, yeah.
Crumbly.
It is crumbly, yeah. So the corals, as the depth increases, they do become more fragile.
Alright. That's about what you need, right?
That is correct. That
Then there's
would
some
be perfect.
little pieces in there too. So everybody gets one. Good portion.
Alright, fortune out.
Guess that's quiver 19, correct?
Years ago.
Yeah.
19.
Something else in this area, then we'll go to the next waypoint.
Yeah, I think after this we can move to the next waypoint.
Patchy coral party or coral?
Which waypoint was this, coral party?
This was the second coral party. There was one where we just after we started the transect, but there's also another coral that I tagged, which is Peccaceres in the way to honest. But
Alright, we have patchy without T because there's less spelling, and then coral. Just wanna run along those three down there to see what we got, or?
I can't read that.
Okay, so we're going to move away from this area and make our way over to another point of interest that we marked off this morning, where there's
21's been taken,
a
yes.
lot of coral colonies as well that we'd like to have a look at.
I just did mark it down on my sheet.
That's okay.
So we've
We'll let you
got
off
that
and start.
coral party. So we don't know what it's called.
One of you two has to change your name.
Spelling was Merida asked if this is spelled that way, so I will.
Yeah, it's an abbreviation of Peccaceres. So I know exactly what that means.
Thank you. Sure. Oh, you wanna go there?
Yes, please. Sorry. Didn't realise you were directing that question.
Can you show the young buck how it's done?
Teach me your ways.
I think she's doing a better job than I am. She's got much more
Absolutely
enthusiasm. Bitter and twisted and old.
Well, I have only been in the sea for maybe half an hour as opposed to 7 days straight, so.
What am I telling her to do? This is how you suck eggs.
When we get to some new coral, I'll get you to work on it.
Yeah, okay. It's much more interesting when there's something to talk about. We have our multimedia correspondent Connor here, taking lots of photos, making us feel really
Comfortable.
confronted.
Comfortable. Do you know what heading you were on when you did this?
Hang on, can we? No, because I wasn't here. It was during my breakfast. Derrida did that one. We were heading towards the west, weren't we? When she would have seen it.
Now I'm going to get my coffee. I'm no fear of cold coffee.
Didn't you make one?
I did.
Oh,
The first waypoint, there was a Pachyceres interest, was a Pachyceres. Okay, cool.
Was it on this mod right here, or was it the other way around?
So it was a big Pachyceres. Karen, you were still here when we marked it.
Oh, was
right at
I?
the start, yeah. It's just a big Pachyceres. I don't think it was at the top of the hump.
let's pull up the photos to be.
Point of interest?
Stuff for that.
Big old bowl.
What's the most recent point of interest? Dagwood is the second one. Coral. Just no. Oh, okay. So it's not. There's another one.
I think you've got to actually put it in the interest part.
Oh, was that one?
Okay, so we're on the hunt for a point of interest, which was a large Pachyceres colony. So the Pachyceres has been one of my target species for two reasons. Firstly, because we expected it to be quite abundant, and secondly, because it is quite easily identifiable. So it has distinct lines or ridges that sort of run horizontally or longitudinally, yeah. Should be in a plating type morphology.
And yeah, there was a lionfish right beside it, so if you see one of those, you'll know we've got the right spot.
Go with Phil.
It seems like the current is turning to starboard, so we may have to do that too over the next whatever hour or so.
Sounds good. How many degrees do you think you'll be turning?
Could well be 180, I don't know yet.
Wow. Alright. Okay. Keep us updated. Let us know.
Yeah, I'll start in 20-degree increments and see how it keeps going.
Okay, so you're gonna start now in 20-degree increments?
Yeah, we'll do that.
Okay, I'm gonna move you to about here.
Yeah, that's no problem. It's not that strong yet, so I'll turn it.
Sounds good.
Okay, so we've just had a question from the unknown explorer as to why I expected the Pachyceras to be here. So before coming, I did do a little bit of research on the common coral species at Ashmore in the shallower areas, and the Pachyceras speciosa was one of those. I also had a look at some literature that had been published on mesophotic biodiversity studies around Scott Reef and also the Northern Great Barrier Reef, and the Pachyceras was also one of those genus, or the Pachyceras speciosa was one of those species that kept popping up and had been found at depths down to about 60 meters. So I had to make an educated guess. And yeah, that was one of the species, along with the Leptoceras that we've also been seeing as well.
Big cushion to start as well.
I absolutely love these big Gorgonian fans. I know our artist Ellie is using those for a lot of her inspiration.
Oh, you did the yawn around. That's classic.
You guys doing over there?
Just kind of big about something.
Missing home.
This is me when he's at home.
It's pretty sad whenever all of us are not working. We spend a lot of time talking to each other.
Pretty much.
Every single day.
Pretty much. Yeah, we do. We do. Okay, cool.
So, Amy, do you know of any bleaching at Ashmore?
Aha, is that it there?
Well, we got. Alright. I was. Is that what we're looking for?
That could be it.
They're both the same thing. Both
Let's pop down and have a look.
Yeah, we'll have a squeeze.
Alright, I'm going to. Should be alright.
Does that look like the photo? I'm not sure it does, but
Let's go these
that's not the photo.
I'll put it PR again.
I'm going back to Ian, he'll be getting another.
That's the photo there. There's a couple of tall pink sponges and a long.
Yeah, don't.
Probably not the same colour, but let's look at it anyway.
Right. That's a birth yarn I have already too big.
Again, could just be at a different angle. Is that? There's two things.
There's two, one tall one
There's a
to
big,
the half
there's a
eye.
big.
And a really large dark
And that's
coral.
fan coral.
Although you could
See
say
that?
that pretty much about everywhere here. There's a sponge,
Yeah.
a big pink sponge,
sort of
and a
a-
sea
That's
fan.
it. The landscapes are getting challenging to recognise once we leave them.
There's two sort of pink
Am I okay to zoom in?
sponges side by side.
Yeah.
Yep.
Yeah, no. One's half the height. It's sort of a good.
It's a nice big colony.
Yeah, could be up this.
Yeah, control.
Monstera.
Yeah, so that's a
You
yeah.
gotta pay out about
Montipora again.
If you focus it sort of
Thanks.
on one of the closer bits, and you can often sometimes see a bit more detail of the polyps. Yeah, something around there. Not a Leptocerus, that's for sure.
No.
Okay. There's a couple of other bits
You
around
guys
it.
could bear with me for a second.
Yeah,
I'm
sure.
gonna spin around. Well, no, we'll do this.
Oh, yeah.
Yep. So this is not one of our species we're targeting. Is there any other ones you'd like
There
to
is a
look at?
bit further down there, I think, that would be more like it. There's a little scully mire there. Cute.
This one?
No, sorry. Let's look at these
Oh.
two here. Sorry, I just got a little distracted with that one.
It's okay.
Aha, that is a
So that
Leptocerus.
is, we've got
And that is two.
one Leptocerus that's got those beautiful distinct coral-like patterns which I love to see. And just zoom out a bit.
What was he saying,
I'm
Cliff?
starting to get the hang of this navigation on the camera.
Hey, yeah, I'm prepared to hand over the reins to you now, Amy.
No.
See, that's the one with the thicker lines.
Yeah. So this definitely seems like the other day when we had a couple of species with the really distinct lines. I think it was the other Ignophilia type ones.
Iconophilia.
Yeah.
What it, slash oxypora slash
That
whatever.
slash yeah. Mystery
But difficult to
species.
tell,
But
yeah. So I
would
think-
you like to grab samples of both of those?
I think so, actually.
Okay. Okay, so which one should we go for first?
Let's go to that one
Let's
first.
go for that one. We'll just zoom in for an in situ, Corey. Here we go, that's the shot.
And can you actually also get a photo of this
What
guy?
are we calling it?
Two.
Leptocerus.
Hyperspectral Leptocerus.
Is it object?
Likely, yes.
Yes, yeah. I was looking at the angle of the camera. I was like, that's
Let's
the
just see on that guy because it's beautiful.
Yes.
Yes.
And can you just take a gratuitous snap of that one, please?
Yeah. Done?
Thank you.
Is that an anemone?
No, that's a coral.
Oh. So this is a little solitary coral.
So do you think you would make it to New Orleans?
So we have for 10
You've
days.
already done it, doesn't matter.
Imagine. What was the
SCOLY.
COLLY? Yep.
That's all we
Agreed.
line up.
Designated driver. Yep.
All line out.
Sorry. Trying to get this more stable.
And I'll
That
add
is
that
fine.
caveat, I think.
Photograph?
We've got the in situ shot already, so you're free to start sampling that one.
And
We can just take a little bit. If the whole thing comes off, we'll grab it, but small piece is fine for PopGen.
Hopefully that little bit that's already been finished.
You something like that?
yeah,
Okay, alright.
because look,
Alright.
there's no way.
Alright.
Unless you want to jump down one. Okay, jump down one.
Alright.
Better.
Yeah, I didn't realize we were gonna have to do that one. Yep, I see it. Gotcha. Okay, grab the literature as well. Amy?
Yeah, we will be.
Alright, so I'll be sure to listen.
Going forward.
Stand by.
Beautiful.
Nope. You're free flying.
That's good right there. Where's your man?
I'm free flying, that's why.
Click off the block because as soon as you're gonna let go.
No, I put it in the hole. So it's spinning.
Oh, you know what? You got 80% level, so when it goes through, it goes from good as it should.
That's all going on. More difficult than it should. Okay, let's find some mollusks. I'm not happy.
Amazing.
Whatever.
Word.
Still looking a little bit.
couldn't have done it better myself.
Literally.
The fire museum. The fire museum. You're making.
I reckon I'd be terrible at both, to be honest.
Oh, you got a big sample.
Alright.
Did you?
No, so we haven't actually gone for it yet. We're just trying
Oh,
to
you're
find
still
a nice
waiting.
stable place. I think
Yeah,
it was a
okay.
little bit harder than what they had anticipated.
ROV.
It's always more challenging than we want it to be. I think if these guys didn't like challenges, they wouldn't still be doing this job.
I think I agree.
This ship from 120 feet. Yeah, something's flying straight down and pull that.
And you're you want to drop it another 10 or 15 meters. Roger that. Awesome. Okay, I'm gonna move your waypoint to about right here.
Beautiful.
Yeah, that's cool. I'll go there.
Go
I
on.
think
That's
Did we
that's the
have
way we
success
want.
with our
Yes.
yeah,
Coral,
okay,
we got that much.
so
Whole
do
thing.
about 15.
Sorry, there's one that's about there.
Stretch
Yeah,
out there.
he's moving crazy, so it's why he's pulling us off. So we
Yeah.
just a second to kind of
Yep, no worries.
figure out where he's going. Yeah. Yep. Which one are you interested in? Which one?
That one.
Alright.
So yeah, you can move
There's some
around to
down here
wherever's
too.
convenient.
Alright.
Oh, there's another one there. Everywhere we
might be a
look,
bit
there's
closer.
another coral. Oh,
Okay.
yeah.
Oh, we're in luck today.
Yeah, I'll just zoom in on it quickly. It does look like a lepto, doesn't it? Yeah, let's grab that one since it's in front of us.
Alright, that one we might be able to reach.
Oh, from where we are.
Yeah. Did you get that?
Do you need an
Works
in
out.
situ shot, Corey?
I will do, yeah.
Should we just let the dust
We'll
settle
wait.
for a second?
Yes. Sounds good.
I'm sorry, I'm getting angry. They want to grab that one right in the middle of the screen. I don't know.
There's all these photos of people doing this.
Yeah, yeah.
Shocking if you
Hair going short, it's like constant all day doing this.
It's super glass.
Guess it's a way to avoid double chin.
I can porch out a little bit more. Alright. Did you get the picture yet or not?
Not yet, we're just waiting for the dust to settle. That's looking good.
Yeah, looks good. It is a bit dusty, isn't it? We get an in situ shot for that one corner.
Leptoceros?
Yes.
You want to do the whole colony as well as the close-up, just so we can sort of see how big it was.
If you two aren't in a hurry, it's the next step move. And probably like less than mine for us just right now I think it's strong side to delay. As soon as he gets in a better position, I probably need a little less. Yeah, yeah, good enough time.
So we just get another shot for the full colony.
Yep.
Awesome. So that's quite a small colony. It looks like it's going to just break off in a little piece, so might be a good population genetics sample. So just
Little piece.
a wee bit.
Going in forward.
Famous last words, a small piece.
That's it.
I think we should just be using reverse psychology on these corals to tell them we're going to take a small piece and then a big piece will come off.
That's it. It has
And
been
tell them we're going to take a
happening
big
that way.
piece and then we'll get a small piece.
Oh,
Oh, there's rocks there.
yeah, there's a big rock right that whole rock there.
It's a really nice colour. That colony up there.
That's the Montefiore, isn't it?
I'm not sure.
Is that? Disoriented, man. Oh, there's another one there.
It's all starting to look the same.
You want the whole thing, right?
Whole thing, right?
We can do. If we end up getting the whole thing, it'll
Yeah,
also be for ecophysiology as well as population genetics. So
Okay.
whichever way the cookie crumbles.
Stays in one
The pancake crumbles.
Yeah, coral crumble.
That's,
Got it. That's
Beautiful.
gonna have to go into.
that's definitely a bio box, that one.
It will. So we already have a Leptoceros in both the
left bio boxes, so we might have to put that in the H.
Bio box H.
That's correct.
I think the reverse psychology.
Yeah, we'll try that for
Yeah.
next time.
Yeah.
Next
It
time we
really
want a small
does.
sample, we'll tell it we want a big one.
And then give us like a little pineapple half group, so we know we want to
Yeah, okay.
say pineapple.
A special hand signal or something.
hand
Yeah,
signal.
yeah.
This is probably one of yours, Amy.
Correct.
Yes.
Yep. This is enough for everybody.
That is enough for everybody.
You want to go that first 16 and not go away?
Nerida hates it when we fill the bio boxes with corals. Just as well, we don't want another one of those starfish like yesterday.
Oh, it was
It was-
so big.
It weighed about 2 kilos, that starfish. Yeah.
Yeah, so the starfish that we sampled yesterday, the big one, is potentially the heaviest.
No,
No.
That wasn't
no,
it?
no, no. We
No
weighed
potential
it.
of that?
It was thought it was, but
It was way off the
Oh,
mark.
I think it was 5.8 kilos. It was the largest or the heaviest for that species, and this one came in at 2.85.
Oh, so we weren't even close.
We were
Wow,
half, yeah.
that makes me wonder
Imagine how
big
big the big one was.
the big
Yeah.
starfish was. Yeah, wow. Do we want to have just a look at what this is? Not for
I
sampling,
thought
but
we
for...
did have a look at that. That's the Montefora.
Oh, was it?
Yeah.
I didn't remember it being so green.
Just a different angle on it.
Ah. Do you want to have a
But this guy,
go
pretty
around?
good. If we're in a comfortable spot.
Are we
Are
settled
we
down
comfortable or
Yeah,
are we?
you're fine.
Comfy
Yeah,
area?
okay, let's have a little piece of this
If
one.
you already want to start calling us and slowly
Start following it slowly.
Yep. Thanks, man.
Yeah.
That's one of those darker ones.
Let's grab a huge piece of this one.
Yes.
This big.
In situ.
Sarah's Pop Jam.
Thank you.
Yes. We're hoping it's just for PopGen.
It's probably not big enough for even if the whole thing
Yeah,
came. It's
that's
only a
it.
little one,
Yeah.
isn't it?
Alright, let's go up for a second. And then I'm going to move a little bit, just so
Okay.
I can change.
What species was the starfish?
Oh, jeez. Give me a second. I'll get that for you.
Someone's asked in the chat.
Where was it? It doesn't matter if we're closer now. Call it a business. It is. Chlamydia catalyi. Chlamydia catalyi.
So we've had a question about the large starfish that we sampled yesterday. So the species is
Chlamydia catalyi.
Thromidia catalyi.
Catalyi, I guess. Catalyi. I will Google that and get it. It might have a common name.
Thromidia catalyi. So I think there's both of us are struggling to pronounce that scientific name.
And that was courtesy of Oliver and Zoe at WAM.
Yeah, so that identification came from the WA Museum from Oliver and Zoe, two of Corey and Nerida's colleagues there.
And we need to do a little bit of checking, but it's probably the first record for Western Australia for that species.
Yeah, so that sample, the starfish we had yesterday, that's potentially the first record of that species in Western Australia, but we just need to investigate that a little further before we confirm that.
Katala's sea star is the common name. Katala's sea star.
Katala's sea star is the common name for that one yesterday.
Marry me, take me with you. There she is. Yep.
Yes, that's the one.
That's pretty fun right there, actually. Brown tips, sort of these characteristic brown tips on the end of the arm. Seems to be a...
Yeah, it was a very pale pink, almost whitish colour, with these distinct sort of darker brown tips on the end of the arms.
Unfortunately, being the size it is, it'll probably, once it makes its way back to the museum, we'll have to go in our drum storage, which is fine. It just means it won't be in our normal wet collection where we house our jars. It's just of a size. Our biggest jar size is around 2 litres. We do have pails at about 5 litres, but obviously this giant sea star will not fit into that, so it'll go into a bag, which it's currently in now, into a blue drum and into some racking in our bulk drum store, which is air conditioned and humidity controlled, and will be data based, and when it needs to be viewed, we will pull the drum from the collection and have it ready in the laboratory for whoever needs to view the specimen.
Fantastic, thank you, Corey. Okay, so we're just trying to set the ROV down in a suitable and stable location before we collect our next population genetics sample.
Get a shot at it, or are you gonna shot it?
We got an in situ shot.
Okay, cool. So go for it.
Yep, go for it.
Down 62 degrees.
So it is a very small colony. Yeah, so yeah, definitely a pop gen sample. Oh yeah. So we're trying to reverse psychology of the corals. So yeah, we want the whole thing. They seem to be breaking off in little bits when we want a large sample, and the whole colony seems to want to be collected when we're just after a small tissue sample.
Well,
You know, they knew what we
it's
were doing.
quiver size.
Yeah, they knew what we were doing.
That's fine, that's Cooper size, so none of the sample is ever wasted, so that will have a tissue sample set aside in ethanol for DNA sequencing for the population genetics study. There will also be some tissue samples taken back to the WA Museum, and also a skeletal sample which will be used for taxonomic identification firsthand. So which quiver are we popping this into? 18.
Yeah, I'll put it in there. So you might bring a boy? That was 18, was it?
It was, yeah. Cool, another successful
It
sample.
was a pop gen, wasn't it? Oh, no.
Just put it as a regular collection because it's probably enough to get a couple of small tissue samples for the museum as well.
Yeah.
He's youngest, but I think that
around there.
Did we end up making it to the first waypoint? I'll just see
Yeah,
if
this
there's.
was it.
This was it.
Yeah, I think that was
Okay,
the one
so
that we identified.
hiding around here somewhere is the Pachyceras, or was it?
That was
Did
the-
they
that was
think
the-
that this one was
yeah, this
it?
one was
Oh,
definitely
okay, so it wasn't actually it.
You want to look around some more for it?
Just have a little scope around to see if there's any other Leptoceras colonies we can sample. I think that's all for this area.
I think I ate too many.
I'm just gonna look at this colony here.
Sugar crack. Colonies do things.
No, so that looks kind of Monticora-ish as well.
So that's the-
Okay, so we can probably go for a little explore,
Okay.
I think, yeah.
That'd be good to cool down anyway.
So I'll set you up straight away. And you're free to. I'm going to get
Okay, so we're done with sampling the corals in this little area, so we're just going to take the ROV in a bit of a wander.
pretty cool picture.
Got it. Fun background.
Nice little fish.
Okay, I guess while we go for a wander, I can talk very briefly about this ecophysiology study that I'll be doing. So what I'm wanting to look at is how these corals are feeding in the mesophotic depths, particularly over a depth gradient from about 50 meters to 70 meters as well. So I want to know if they are mainly relying on autotrophy, so obtaining their energy from the symbiotic zooxanthellae within their cells, or if they are also relying on heterotrophy. So it's usually sticking out their polyp tentacles and grabbing zooplankton from the water column. So I'll be investigating that using an isotope analysis, looking at certain isotopes of carbon and nitrogen within the tissue and also within the symbiotic zooxanthellae. This can also be complemented by fluorescence readings, which I'm taking with a PAM fluorometer. And we're also fixing some decent tissue samples for histological analysis back in the lab. So there I'll be looking at what kind of nanostructures are within the polyps, how that might change over a depth gradient between species and also between conspecifics at different depths. And we'll also be on the lookout for any signs of reproductive cells, so any oocytes or spermatocytes as well.
There's a inset shot of the large sea snail you guys collected.
It's up on live stream now.
Oh, cool. So if you just look in the corner of your screen, there is a shot of the starfish that we collected, or the sea star we collected yesterday.
Catalan's sea star.
Okay, so there's a nice. Is it easy to set down here? You can see 1 or 2 potential colonies. Cool, so we're at 59.4 meters depth at the moment, so we have gone a little bit deeper, which is really good for me because getting samples at different depths is good to analyse how these changes in physiology occur across the depth gradient.
Oh, you use cameras. Yeah. I don't say that one to the left, sorry.
Oh, where am I going?
I got you, that's move to the left a little bit for you so it's better. There you go.
Okay, could get an in situ shot of that.
Sample or object? Oh, sorry. Yep. Done.
We will see how much we get.
See, Leptoceras?
That's it. It is Leptoceras, correct. So if we could take a sample of that one, that'd be great.
We're gonna go for 16.
Okay.
So,
How'd you gotta do that?
So, Amy, with all these samples you're gathering, what do you think time-wise you'll be in the lab for tonight? You've had some late nights on these coral collections.
We have had some late nights on the manic coral days. I don't know how I thought I was able to pull that off all by myself with even more samples than we had been getting, but I'm very lucky to have had the other students on board, Declan and Chloe, assisting me in the lab on these crazy coral days. So how many samples do we have at the moment? It's probably got about 7 for ecophysiology, so that's definitely a few hours of solid subsampling, but that's completely fine. I enjoy the late nights, I'm not gonna lie.
And it seems we're rotating each day. One day's a full biodiversity day of invertebrates other than coral. So I guess the museum team are quite flat out on that day, and then the following day we go to shallower water and we do a big collection of corals for Amy's and Karen's project. So they're generally in the lab quite late, and the museum team have a shorter night. So it's seeming to work quite well as we make our way towards the end of this trip. And yeah, I guess it's your turn today.
It is, yeah, it's pretty good it's worked out that way. I mean, while the museum team were in the lab till fairly late last night, I was watching the beautiful sunset on the back of the ship, which was a nice break.
Found another Leptoceras, I see.
We did. We're just sampling it now.
Great. Have you found that Pachyceras we were actually looking for yet? Not yet.
No, the colony that the crew pointed out that was marked as the Pachyceras was just the Monticola.
Oh, okay.
So we've gone for a little wander. But we've got a nice Leptoceras and it's at 60 metres, so
Oh, cool.
that's always good news.
That's because the tide's coming, not because we're deep. Is this just a pop gen sample?
Yeah, I think so. So you happy with that for your
That's,
Cool.
if we can get it in the quiver, is a perfect size pop gen sample.
That's it. Happy with this piece, or did you want another?
Oh, I'd grab probably one of the other little ones just to be safe.
given it's broken.
This would be a population PopGen sample.
PopGen,
Yeah.
sure. Looks like 16.
It is.
Alright, we've got a big piece.
The whole thing.
Hole going in.
Oh, actually, I guess there's a couple of pieces there, isn't there?
Can we go back and get that other piece?
If it's easy.
Yeah.
Yeah. I think there's a
It's
little
on the
broken
ground.
off piece of it.
Yeah, it might be good to start to move down to the other coral party at the other end of the.
Sounds good to me.
Oh yeah, that piece would be perfect.
So we've got another coral party on the point of interest marked out.
Yeah, it's more towards where we started the transect, which is the other end of the spine. Fantastic. But there's also a couple of sea cucumbers that Nerida might like to have a snorkel at. Go past them.
The museum sponge guys are watching and they might get back to us with that. I'll chase that up with the bus.
Yeah.
I'll just someone wants to know when people run sponges. I don't know how simple that question is.
Happy with that?
Yeah.
Beautiful. Still need to get a couple more packies.
Yeah.
And
Yeah.
Settlements.
7, 8, 9, 10, 11
Yes.
PopGen. So we need another 5 or so PopGens. Minimum of 15 PopGen. One, two, three.
We've got 12. Up to 12 left over.
But this one I
I'm
think
not
is
sure
the,
if they're-
I'm not counting
Oh, a
that
different one?
because it's, that's actually a taxonomic specimen.
Ah.
So it was the one that was really wavy.
Awesome.
So we're going to do one.
Yeah, so we can go to the point of interest where the other coral hotspot was.
There was a couple of
Some
sea
cucumbers.
cucumbers I think that we marked.
Oh, see, alright, yeah, we're going to move. Alright, gotcha, gotcha. Alright, that'll be a bit of a trip.
It won't take that long.
So yeah, let's head in that direction.
Alright, so roughly waypoint 2.
Start with the ship first.
Yep.
Updated waypoint, right?
I'll be there. in a few minutes.
Scholarship.
I'm going to do a concept global pandemic totally put crap on.
My last one was Sublime Offspring.
This one?
That might be a hard spot to stop though.
Yeah.
I had a girlfriend cheated with a guy who looked like the
Let's just go a little bit further down the transect.
association.
Wow, it's an amazing sponge.
Yes, sponge. Pretty cool.
So I wonder if it talks about you.
What song was that?
I'll play for you.
Probably won't like it because it's all shit song.
The lead singer?
Yeah.
Do this ship to shore thing. Are you
They were already
there?
famous before she did it. So.
Just keep collecting. Having lots of fun. I think Nerida will come in and co-pilot in a minute.
No.
Sounds
She was the
fun.
one. She's gonna make it honest man out of it.
She's here.
You're engaged. If you are, that is.
Are these instructions to me? Or we were just counting the samples, so there's a couple of cute
The worst part,
waypointed,
the worst
so we're
part
heading back in that
was
direction now.
we were learning that we broke
And
up,
another Pachyceras
and
and
I got the cheating box.
another coral pond. So I guess
So I couldn't work, and I was broke, so I like sleep in my car for a few days. The cheating box. So I'd be wearing a fish. That's called yeah. So I live in my car and she gets everywhere. For the chicken box. No, it's scary.
We go.
So is this so I don't know. Do you want to work offshore like this when you graduate? Want to work offshore like this when you graduate?
Sure.
Like, do you want to do like science cruises and all that all the time?
Yeah, I would love to do science cruises at some point in the future. I'd love to get into research, so it's a pretty difficult area to be successful in. So we'll just have to wait and see, but this is a lot of fun. I've had a great time this morning. It's always good being out on the open ocean.
Yeah, just double check. It's from Stanford fast, it's really well.
I think I'd have to have a much better routine though if I did this longer term. I feel like I'm eating, working, sleeping. Repeat.
Yeah.
Yeah, that's our life.
Yeah.
It is our life.
Are you looking for mollusks there, Amy, as well? Or?
So yeah, we're going to a waypoint where there's a lot of corals and there's also some sea cucumbers, which will be Nerida's interest.
Too bad we filled all the bio boxes already.
I know you're teasing me. I'll just put them on top of the
corals.
corals.
Yeah, but hey,
Didn't even think about the question.
thanks, sir. We're moving locations right now, so we're kind of going towards the ship. Just to let you know.
If you're happy.
Thank you, sir. We'll speed up a little bit as well to get to there again.
It's a cool little
Yep, that sounds good.
weird
Man.
looking sponge.
If you stop for something, we'll let everybody know.
Yeah, sure.
Like a big Milo. This is Milo territory.
Yeah, it's be lovely. We're looking for a big snail, please, guys.
Big snail, right?
Have you spoken about your white whale?
About the size of a gridiron football. Is that a good comparison for you?
By the what?
A gridiron football or
A real football.
American football?
A real football.
That would be a
Not a Canadian. American football.
Come on, Milo.
Yeah, where's good?
Do you have a celebration protocol if we find him? I love it. Just chaos.
Screaming. Screaming with joy.
So I figured out my
How like
my roommate's cousin.
We're doing good. We went to find the March Pachyceras, but it ended up just being Montipora. So if we could get a couple more, or a few more Pachyceras, it would be great. We're doing pretty good for
She
Leptoceras.
let me know that the roommate I'm actually friends with is considering moving to Hawaii. And
Yeah.
stuff is just her and that dog.
12 so far. That's great.
We've got quite a few for,
And none of the lined ones, just the other one.
yeah. So there was a weird one that we got samples of that had really dark lines and quite a dark central
I think I saw that one
I
actually.
think that was
It's more
a
orangey around the
juvenile of another we saw the larger colony today and we think it might be like an Echinopora Acrophilia. It was really brittle and really hard to work with in the lab. It basically just crumbled but still stuck together.
Yeah, yes, with that sort of tissue.
Yeah, like the film, yeah. So that was the
Pretty
easiest to
helpful,
work
yeah.
with in the lab.
Sort of dead shell.
They're a bit fired up for violets again today because there's so many soft corals here.
Yeah.
Pretty good pickings, I reckon.
So you said red beans and rice for lunch?
This is a little solitary coral.
You want to look at it?
Sorry?
Did you want to look at something?
It's alright, we'll just leave it.
Yeah, only stuff.
Might stop. That might be
Yeah.
one as well.
Which one?
Just here.
Will this be there?
Yeah.
But I'm not sure. Do you know the species that can also conform?
I don't. It's one I'll have to ask her, definitely.
If she's in the library, she'll be.
I think she's doing the ship to shore
She's still.
thing at the moment.
Right there?
Yeah. Yeah, I know she has collected a couple of these. I'm just not sure how many she wants. It does look like a Cycloceras.
I don't think that's the one she's after. Eh?
Yeah, she got about
Portobello
maybe
mushroom.
2 of them.
Oh,
Yeah,
okay.
it is a portobello mushroom looking thing, doesn't it? I'm not sure if she wants more, but just I think she's going to be stuck
If everyone
talking
gave me
about
about 5 meters. Want a friend, Brad? Thanks, sir.
Sure, yeah.
Yeah, can you just write in
I can just.
the word comments Cycloceras or solitary round coral?
Okay.
In the events bit.
We can put a point of interest, right? And,
Yeah. We'll just POI it.
Alright.
I don't think she'll want to.
oh no, it's thumbs down. Yep.
No, okay, thumbs down. So we're not needing to sample this one.
We should have an image of it when we did.
The solitary one. Yeah. Cool. We can keep going.
Alright. Yep. That's block point of interest.
So Karen doesn't want to sample that one, so you can probably put a comment now. That's fine.
Unneeded point
Yeah.
of interest.
Let's say it's an unneeded solitary coral.
Done.
This is definitely a great habitat for some of those sand-dwelling mollusks that we're after, so just keeping our eyes peeled for any gastropods sort of popping out of the sand as we move towards our next waypoint.
sea star, is it?
65.
Or is that a small
Whereabouts, Corey?
to the right extreme?
It's a sea star.
Oh, a small one of those
There's,
Cadillac.
yeah, I think there might be a small catalyst.
Yeah. It's
Round tip.
Let's do an image maybe.
I'm just gonna have a look at this sea star to the right. Very similar, doesn't
Think a
it?
juvenile?
Oh, that might.
A few more speckled than that.
Yeah, I'm not sure if it's the same or not. Could just be juvenile.
Oops.
Wrong way.
Backwards. There we go.
Is that like a smaller version?
Oh.
You went?
Oh. We actually put one up on there. That was I've got the one with the elite tail.
You want? They're about to come in with the camera too, so they're gonna be shot.
Get that elite tail and see if that's
Alright, I guess I can wait.
right off to it. So we should throw it on something.
Did we want this or not?
No, I think they're just having a nice peruse of it. Yep. That's
I
cool.
will just wait. Just
Hang on, guys. Can we just get an image again?
Sure, sorry, the
No, it's okay, don't
ROV's
worry.
moving.
We'll have an A snap. Don't
I think
worry.
it's getting pulled up.
Oh yeah. I'll do a POI then.
Yeah.
Or
I can go back down on there if we can.
If you just do a video on it now, it's fine.
I can go back down on there if we
No, all good. Keep going. Keep going.
yeah. Done.
Is that where you?
Yeah.
Or is that something else?
Yeah. Oh no. Are they going
No, no.
the ROV? I think are we going to
No, oh, that is
I can't even tell. So where are we headed?
Is that, it says CQ2 over there?
Oh, that one.
Yeah, Darren, I changed my mind. We can pass out about 10, that'd be great. Thanks, boss. I was wishing I drank coffee this morning. I was exhausted. When I was doing at the gym, I was like,
Nope. Do you feel groggy?
I felt not groggy, it's more just still really tired. It's like,
So I think the autofocus is out. Oh no, there we go. All good.
yeah, I missed
Is it not
a
auto?
couple meters off the sea though. Whenever.
Okay. But, yeah, it's good.
Go all the way out there. Like tissues. Straight. That's the straightest he's ever gone.
Yeah.
Ever seen. This shit that straight. Of course, it's not where we wanted to go. What are you teeing down here? Tell me, right?
Oh, it's this moving thing.
Oh,
It just
yeah,
it just
have a look.
moved. Hold on,
Yeah.
let's just have a look at.
Might be another one of those little pipefish, maybe,
Yeah.
or some sort
Striped
of
looking
fish.
Yeah.
It's a little fish.
Yeah, I think it's like a little blenny fish.
We can keep going.
Keep going?
Yeah, thank you.
It's lucky we don't have a fish person on board, otherwise we'd be stopping way too often.
Great concept. Very nice. Wow, dear.
Interesting that dark octocoral is sort of multi-stalked here, whereas previously it was just like a single stalk.
I missed it. You can keep that. It's a little Leptoceros.
You guys did that, this transect east or west to east, right? This transect, you did it west to east.
The opposite way that we're going now.
Oh, yeah. So I'm gonna try to come to the west and go back east and find it.
Doesn't matter.
Just realised we were both still at lunch.
Do we assume that others are already doing this?
They
So that's
Do you guys want to go? Why don't you just go?
Well, my instructions are that Karen's doing a ship short and coming on to then swapping out with me. But if it goes really late, I mean, so I can take over this and you can go and then swap back for it.
Yeah, sounds good.
cool, good plan.
If it gets a bit late, I can just swap with you again then. Especially if we're in coral territory.
Should we just?
Yeah.
Do you mind doing a bit of a cut run?
Can you manage three?
Manage things.
What's that?
No.
Sorry, I got my eye.
As if I've never done that.
So Nerida, with the mollusks, we target shelled mollusks as well. Ideally live animals, but we also do take shells, and that's for mollusks that's quite advantageous because it can indicate that the species is still living in the area. I just wanted to see if you wanted to comment on that. The value of shells, a lot of people wouldn't probably understand that, but for us it can be a record, and depending on the condition, the shell can indicate the species is still obviously in the area.
Yeah, so I think it's important to remember too that many mollusk species are actually based on the description of only a shell, and so we don't have information often about the live animal. And why that's important these days is that we, of course, would like to use DNA sequence data. It both helps us resolve relationships and what species are more robustly, but it also allows easier comparison for people comparing their fauna to the animals that we've sequenced. So it's a really great bridging sample if we're able to get something that we can identify the shell and link it to a description of a shell, but also get that additional information about the live animal and tissue for DNA samples as well. So it's sort of a good bridge between the old and the new ways of doing taxonomy.
So at the WA Museum in the mollusk collection, we actually have a huge dry collection, which is essentially a shell collection. About 2,500 trays of shells and around an estimated 350,000 lots of vials of specimens. So that's been building for a good 100 plus years now. But we have quite often now, as Nerida's mentioned, we're doing molecular work, so we've used the record of a shell to, and where it's been found to then go revisit that location and do some targeted collecting where we're trying to find live animals. And so it really is a fantastic starting point to look for certain species. If we've got a dry record or a shell record, we know the animals likely to be in that area. We've got generally a GPS point depending on when the sample was taken with technology, and we quite often we'll go back to those locations and put in a concentrated effort to find a live animal, so that we can proceed to the genetic work as Nerida's talked about.
Thanks, Corey. Corey is one of our invaluable technical officers at the Western Australian Museum. He's worked there for a very long time and knows the collection and knows the animals incredibly well, like most of the technical officers there. We're really lucky to have their knowledge.
Something coming through inside.
It's fine now. It's fine now.
I can hear it in the background all the time.
It's fine now.
I think they're doing an outreach right now.
Yes.
Yeah.
I'm looking at the abundance and the diversity of soft corals and noctocorals on this site. I'm really excited to do a bit more searching for ovulate or spindle cowries later on today. Although of course I will have my nudibranch eyes on. But at the moment we're just making our way to a point of interest that was marked earlier. We have to leapfrog over these big beautiful fans as well.
I guess they were looking like this, maybe. This is like a really, really large.
Yeah, do we have an image up for the point of interest that we're going to?
It's just the
Snake! Snake button!
the coral party, is that what we got?
Snake button! Snake button!
Snake.
Can we just try and get a close look
We
at
don't
him?
need
Yeah,
sort
just
of-
trying to
Okay.
avoid that big.
Cheers.
Hey, Rick,
Yeah.
Can you pair up on
If you
some
need
meters?
a depth, I'll just put that.
Looks like an olive.
So I should only press the snake button once, eh?
Correct.
And then we'll know to get the frame grabs around it.
Yeah.
What a beauty.
It's just amazing to see them move in the water. It's hard to remember that they are actually air-breathing organisms. Gorgeous. Okay, I think we've probably got a good...
You're like chasing this thing while trying to avoid corals.
It's an exciting ride. But I know how much you love snakes and you'd prefer to look for a sea cucumber than a sea snake, right? I have a friend in this mission here. I always find when I'm scuba diving and see a sea snake, they remind me of how ungainly and how awkward we are in the water. Their movements are just amazing. So seemingly so effortless and graceful.
When I hear you talk about scuba diving and seeing the sea snake, I remember why I don't scuba dive.
To be honest, Chris, I was never very comfortable with sea snakes, but I think the last trip we did to Exmouth Gulf, I kind of finally got my game on. Yeah.
Yeah, the first time I went diving with a sea snake was at Scott Reef, which is another a reasonably close reef from Ashmore, maybe, I don't know, 300-400 kilometres south, I'm not sure. But I dived into the water and immediately the snakes made their way towards me out of curiosity, and it was just the most
Confronting,
bizarre, but in a funny way, relatively relaxing kind of experience.
Not the words most people
swimming
use.
around your mask, and
Is this the point of interest?
so it was a great experience.
Okay, here's a cuke. Let's see if he's our point of interest. Well spotted. Fantastic. Right.
So do you want me to?
We'll just settle down and have a look at him. Yeah. Alright. So maybe just get a frame grab, but we'll have a look. Cool. Oh, there we go. It's a beautiful looking animal.
Oh, morning, bud. Yeah, absolutely. Let me see you over there.
Any yellowbits?
Just having a bit of a scan for those little parasitic snails.
What should I call this one?
I don't know.
Just call it Fury.
Okay, so would you mind giving him a little gentle tip over?
No problem.
So we're just scanning this animal for very tiny parasitic snails called eulimids. So they can occur on the outside or the inside of the animal. So we're just looking for any obvious signs of them being on the animal, and we're just gonna, sometimes they're on the underside, sometimes they're on the top. So we'll just have a little flip and see if we can see it. It's a stunning animal for a sea cucumber. I actually think they're lovely animals, but not everyone agrees. Hi, Merida. Hi. Oh,
Yeah, we can see you guys.
Excellent. Pleased to see my cucumber.
Hi. This is Corey. He's also on the science team.
Hello, everyone. This is Captain Corey.
And over here we have the ROV drivers. So these, for people here, they all drive the giant SuBastian, which is kind of like the bus thing that Karen was describing to you under the ocean. It also has these big claws, kind of like a crab, and that's what they use to grab the different specimens when they're bringing them up. You can probably see
Oh,
that
there we
on the
go.
screen.
That'll do.
They're trying to get a sea cucumber at the moment with it.
Oh, is that you?
Oh. I don't know.
I'll roll
He's
over.
flipped. Yeah, that'll be great. Thank you.
So this is remotely controlled.
You want me just to hold them?
That would be fine, yeah. Yeah, because I think he's going to ride himself pretty
We're about to
quickly.
pick him up. Very exciting. So
Yes.
what we do with the sea cucumbers up in here is we will flip them over and we'll look for little parasites on them. And a lot of the parasites are host specific, meaning that they basically live just to latch onto this cucumber, which is really exciting because it means that most of the ones that we find are the first time we've ever seen them. But it doesn't look like there's much going on. Sheridan would be very sad now.
Well, you need to have a proper scan of his tummy. So we're just getting him in position. Just another day in the office.
They are pretty
So this robot is operated, so they use their hands to move this little controller, kind of like you're playing a game control at home, except much more sensitive. And then when they move their hands, it gets transferred down these, what's the kind of cord is it? Fiber optic cord, and it moves the robot's hands at exactly the same time, which is pretty cool. Connie will show you.
Does anyone have any questions for us while we're in here? Any new types of coral? I don't think we've found any new species. We haven't been able to identify that really cool one that we showed you before that had all the pretty colours. But this is kind of the first time that we're looking at this reef, so in that sense, it's everything that we're seeing for the first time, I guess. So not the first time ever, but certainly the first time here at this sort of depth. Thanks for your question.
Thanks, Ibrahim.
new alignments, but so we're not probably going to predict this one as we see something. So that's quite a big one.
Yeah.
Wow.
Yeah, that's the one.
Which biobugs was that?
My favourite type of coral that I've seen is
I think
definitely
maybe this one.
sponge coral,
Okay.
which isn't everyone's favourite on me, but it looks like they're these ancient kind of vases or vessels that have fallen down to the bottom of the ocean, and they're massive. I'll definitely show you some photos of those later, or we might even come across them. And they're really big. Actually, we might be doing a 3D print of one, which is pretty exciting. We've got a 3D printer on board, and I'm working with one of the guys here, Jayron, to maybe make a print. It's really cool.
Thanks for your question.
What's your favourite one, Chloe?
It's pretty hard. A lot of the coral is quite brittle. So if you've ever had like peanut brittle before,
Oh,
if you
it's
grab
sticky.
it, it can just kind of
Yeah.
shatter.
He'll-
But we
he'll
have
release,
such
yeah,
talented
there we go.
technicians here that they are doing just an amazing job at breaking us little bits off. Or sometimes they kind of get
Yes,
underneath
correct.
it and like pop it out of the ground.
It's almost like they're pulling
Yeah.
a big one of those giant mushrooms out of the ground.
Yeah.
If they get the bottom bit, it'll all come out. But if they get the edge, it can break off a little bit easier.
It's not easy work for sure. They're very talented.
And it gets so intense when they're doing it because everyone's sitting on the edge of their seat.
Yeah.
High concentration.
Cool. What's the next one?
We're going to keep walking through.
Number 2 or 1. That was sequence number 2.
Okay. Yep. Cool.
Another sequence.
Let's proceed. I don't know if they're the same or different. I wasn't the one who marked these.
I marked it.
Oh, did you? Are they the same? Let's go have a look. We can look.
It's along the way. You happy just to kind of
Yeah, absolutely. Yep, absolutely. Thank you.
And that one closed because it were.
That one's taken?
Would you like one?
Broken lid.
Oh no, thank you. All good. I'm a still person.
That one, yes.
Now on the way, we're going to be keeping our eye out for Pachyceras, that sort of long. Yeah, you know now. You know the one.
Yeah.
Apparently, I may have flown over one before without mentioning it, without noticing it, so I'm in the bad books. Must redeem myself by finding another Pachyceras.
Hey, Russ, can you bring us anything, please?
Coming in 10.
If you just look down here, it's a sea cucumber marker.
You're right.
Big fan. I'm a big fan of big fans. Sorry. Couldn't help myself.
Okay, that's up 10.
Thank you. Probably would have been connected if they were probably on the translate.
Yeah, I put that marker in there somewhere.
Which way were y'all facing when y'all were going?
We kind of came basically.
Were y'all lat on it?
No, we came over the top of it like I am now.
Okay.
What I might do is come to the south and come to the west, then come back on it. So I guess these corals are so high, it could have been on both sides.
We've got an image of that point of interest, and it's in between two fans.
In between two ferns? Roger.
Great show, great show. Yeah, it's definitely a different species.
What color is it?
Chestnutty, browny, purple.
Red.
Rougey, reddy, browny. Sort of a little bit similar to the fan, the color of the red fans, and it's spiky. Oh, that's it. Yeah. Good job.
Oh, that big guy.
Oh, the spikes. I think it's a different one, actually.
I think it's the same one.
Really?
From this side. Yeah.
I think it's a different individual, but it's fine.
Maybe it's just sleeping.
He's right where I put the markers.
Oh, okay. So his little points are a bit droopier now than they were earlier today.
There's possibly another one right there. Standing set. Bottom up. Set. Or no, just the fan. Yeah. Right. Just trying to get over here so you can get your arm out. This is what they're gonna roll over.
You heard my little bell.
Facebook's most of the longest time was 104 hours. The longest dive, 104 hours. I've had ROV in the water for 28 days.
Hi folks, we've just been swapping everybody in and out to grab lunch, and been a lot of activity in the background, but yeah, full tummy now and I'm back ready to go. I'm Dr. Karen Miller. I'm from the Australian Institute of Marine Science. I'm the chief scientist here on board the trip, and so I'll be talking you through some of our coral collections for the next wee while. We're going to head towards that next waypoint, which is hopefully Pachyceras.
Patchy coral.
Good job Nerida just left the room, because there's another sea cucumber there. We can just swim past that one. Pretend it was never there. Our bio boxes are full anyway, so it's all good. So we're just cruising back now along the transect that we ran this morning to check out some of the interesting things that we noticed on the way past, and hopefully we're making our way back now to a coral colony, which is, fingers crossed, one of the target species that we are wanting to sample. It's sometimes hard to distinguish the coral species when you're a little bit of a distance above them and cruising around at 0.1 meters per second, so we usually have to come back and look at them. And even sometimes when we come back and have a close look at them, they're still pretty hard to tell apart. So. I remember that sponge from this morning. It was the kind of weirdly upside down barrel one.
Total deck, go check.
We're going to
Yeah, I got you. Got me?
Yeah, I just changed my bearings.
First stop is just a secret coral, and then we're going to the coral
Right,
party
Charlie.
after that.
we go to the coral party before we take anything. We just do either a crisscross or something so that we can get from all angles, and then it'll look really good to see
Pretty
this
good.
way before we start sampling.
Yeah, good
That would be
idea.
great.
Okay.
And that'll be a nice.
Perfect.
Russ, can you call them up on the phone and actually take a measure of those?
Roger that, on the phone. Ready, job take, come check.
That's perfect, lot of everything.
Come down.
You see any wait for it, I'll start moving there.
Roger that. Thank you, Eric.
Is that just to the right, or the left of that sponge on the right hand side?
That one there, yeah, I was just wondering whether it might be. Let's have a closer look.
I'll swing around to the facing it south so I don't hit these sponges, but
That sounds like an excellent
that's
idea.
looking like, I think that's right where,
Because we'd have been coming the other way, wouldn't we? Oh,
yeah, on the back side, just take these sponges.
because that was the bathtub sponge.
Yeah, there
we
it is
saw.
right there, yeah, that's the big one.
Yeah.
That one's beautiful.
That's the bridge.
Should be looking for
Hello. How can we help you? We've got Vite here. Is Vite good enough? Okay, I'll get him for you. MT required.
Hello. Hi.
I have a sneaking suspicion this is not the coral, so I can just zoom in on that and I'll let you know whether to...
He left 45 minutes ago, but we handed over. So
Nah, it's not the right one,
He might
but
be having lunch,
it's certainly not a
downstairs.
pachyceros.
or
No, it's our Echinophilea cipora thing. So no, that can't be the right one.
he's
yeah,
not
it
down
must
here
have been a
anymore.
little bit before here, I'll just back up. I think
Okay,
it's-
you're welcome. Bye.
Thank you, Kevin. There it is.
That looks better.
I'm just gonna approach it from the other side.
All good, thank you. Yes. It's a pachyceros.
Yay!
Now, are we using reverse psychology or shall I tell you we want? We would like a Texas size, I think was the reference earlier. A reasonable sized piece, please. Although, preferably three small pieces so it fits in a quiver. Or in 3 large chunks, quiver sized. Because we've run out of bio box because somebody kept putting sea cucumbers in it.
So this is just the 3 rings. Yeah, so you can just search the label of this one you're going to say they're all the same skin, like multiple measurements. No, I can send it to you. It's just a really rough sort of data collection for this trip. So if you, I don't know where you want to work, so you can just add more of it to the
Can we do the in situ photos just before we get carried away? Sorry, over enthusiastic and I forgot.
actually I think I'll probably
In situ photo, please.
Yes.
Thank you. Can we zoom in any further? Or is that it? Oh, it's not going to help anyway, is it? No, that's cool. Thank you. Oh, we brought out the big guns today.
sorry, it's really slow.
Net control comms check.
Yeah, got you, Ben. You got us.
Roger that, lovely.
That's a perfect size piece.
You want 3 of those? Or something?
No, that I think is probably enough. We may want one more bit, but I'll just decide when I've worked out what size it is. What is it? That's about a bit over 10 centimetres across, isn't it? We might need another piece that size.
Okay, I got it.
Is it a broken up? Yeah. Also, we have to do that empty arrival forms today. Yeah, just.
So we'll take multiple pieces of this colony because we're going to use them for a couple of different projects. We'll use them for the population genetics, we'll use them for taxonomy, we'll use them for ecophysiology studies and also reproduction. So we'll take a few pieces of this one. The good thing about sampling corals this way is that even though it damages them a little bit, they're able to recover and to continue growing.
You want another piece?
Yes, please.
Like some of the bumping stuff or another big piece?
Another big piece, but we could come back for that as well. So if you can get maybe, I don't know if that piece looks like it's
Yeah, I'll get that one.
doable.
it's more accessible to very
I reckon if we get that bit and that bit, that'll be perfect.
That's good.
Yep, I think that's good, thank you. I might just zoom in and have a look at that other one in the back too before we head off.
So here's
Thank you. Looks like another Pachyceras at the back there. It certainly is. Could be a challenging one to sample given it's so encrusting. You happy to give it a try? Challenge accepted. Okay, thank you very much. We should do the in situ photo before we. Oh, you already did that? I'll do another one just to make sure it's beautiful.
Do you want me to get? I'm gonna get closer anyways.
Oh, okay.
No, I think you're good.
You're good?
You need to
All
get
right.
down to the-
Okay, how's that for an in situ? Beautiful. Thank you very much.
Study.
A pretty colony.
Yeah, trying to move that little guy out the way so I can get it.
I'm liking the size of that piece. It's too big for a quiver, isn't it?
Barbox.
Yeah, it might have to go into H.
Okay.
You said H.
Please. If we can get the cucumber to shuffle over for us. Or the other H.
The other end.
How many are we up to?
We've actually only collected Pachys. So this is our third good sample of Pachyceras.
Oh, that's fantastic.
It's getting a bit crowded in the bio.
Someone asked if it's a nudie under the coral.
It wasn't.
Damn.
Anything else you'd like to hear?
I think that's good for here. If we can head across to that other coral party waypoint.
It's not coral party, it's coral party.
Oh, it's a coral party. Oh, is that an upmarket party? Having a party. It was pretty early in the morning when we were trying.
That was kind of the first waypoint I think we
It was, yeah. It was very shortly after we'd started the dives. It was still quite early. In fact, I don't think the sun was up at that point, so I think we can be forgiven for misspelling. Before we... What are we looking at? Oh, a stomatophore! Oh, look at him! Cool! Quick, photo! Woo. Oh, look at him! Oh, look at him go, it's cute! Oops, wrong way, sorry guys. Yeah. Oh, he's gone in his hole. Oh, here he comes, here he comes. Hello, cutie. Yeah, you can drive it much better than I can, thank you. I just got overexcited there for a minute. Look at those nicely spotted. Is he a basher or a slasher? He's a basher, isn't he? Sorry, do you do that with snappers? Come on, go for another little wander. Wow. Try and collect it.
If you just zoom in a little.
Oh, can I?
We might be
Oh, here he goes.
might not
Come
be,
on.
it might be just a little.
Oh, it looks like a bit of a half one.
It's just the base. Yes, I think.
Is that how I made it, or is it some weird biozone? Sponge or something. One of those things.
Yeah. We do need that top. Yeah, it's silk in the back.
Uh-oh, I feel a karaoke night coming up. Seriously cute.
It's cute as psychotic killers. Very efficient killing machines.
But so long as you're cute, you can get away with it. Hang on, out you come. Oh, here he comes. Here he comes. Yeah.
Okay, let them drive. Let them drive. No, it's just too.
Don't trust the biologist with the focus knob.
He wants to come out.
Yeah, he's testing us to see if we're going to do anything if he moves.
He's teasing.
He's just not really convinced about us. Who parked a bus in my driveway?
Ruined the view. Whoa, don't stop looking here.
No. Oh, that's. Oh, no, maybe. Alrighty, I think we better get on with the job, hey?
That's a little fun.
Were we going to look at something else then?
Do you want to just go around here a little bit?
What I'd like to do is head across to the coral party.
There you go.
Oh, did he? the Coral Partey waypoint. But before we settle down and start sampling, looking at the corals there, if we can do a few fly arounds so that Ian can do his 3D
Yeah,
with
if
it,
you let me
that'd
know
be
when
really
you're doing
nice.
it, I'll check the cameras, and that would be great. Thank you, Karen.
Yeah, we're only about 20 meters away from this.
Okay.
Let me know if the hips are.
How about the structure? What's the kind of structure? Relative to where we are, where are we heading?
I don't know. I haven't been there yet. We're going to be over here. Okay, just a second. Just let me know if the hips are.
Yeah. I'll go and look at that first. And come back. You know, that's a few.
Oh, big
Oh.
potato pod.
Whoa. What is?
Or whatever their real name is.
Yeah, never let two.
Fantastic.
Have we got the? Can we see how big that is? Have we got the green spots on?
Yeah, the lasers are on. They're just.
Oh, I see them. Yeah. Okay. Gosh, I was looking for them further out than that. Yeah.
Oh,
so
wow,
he
yep.
is what? That's just coming up
He will
for.
not fit into coral.
No, definitely not a quiver-sized fish. That one.
Look at that. Nice images.
Yeah.
Woo. Beauty. Oh my goodness. That is an impressive fish.
You found the fish they like.
Fans.
Not bad for a fish. Yeah.
Yeah. Good job. I also like sea dragons.
Sea dragons. Cool lionfish. We saw a couple of those this morning.
Yeah. They're
Yeah, they were pretty cute.
Super big, but
nice one.
Very nice. Okay, back to the coral party. We keep getting
straight. What'd you say?
back to the coral party.
There you go.
Is this the whole thing?
No, I don't think we're there yet.
I think you coined it this morning, wasn't it? Somebody said it, so we wrote it that way.
Oh,
Maybe
okay.
it wasn't you.
No, I don't think it was me. But then again, it was early in the morning, so you know, and it was pre-coffee, so anything's going.
I'm really struck by how few pipelines. There's one on the rock there.
Mm.
Just really
Like you know how deeper we see heaps of sea stars, and here we see
yeah,
none.
yeah. No, it's yeah. And
So
the
it's-
cukes.
the
up
cukes
here.
are up here. Yeah.
Yeah. Wow. Gosh, this is amazing.
There's not quite as many hard corals as what I would have called a partay. So I think we might need to come around the other side.
So I can, let me just, I'm just going to take my half turn out here. I'll kind of do a full round away here.
Just let the shades of pink. Now we're looking more. There's a few big ones up there and a big one there. I suspect we're getting more to the area of interest. Yeah, this is looking better. Quite a few corals, hard corals there. I could zoom in quickly and see if they're the ones that I want, if that works, or are you... Oh, that's right, we're doing the times.
Yeah, if you keep going, yeah, it's great so far. Just gentle. I'm trying to keep the altitude fairly low, but again, keep coming up and over. Yeah, you're doing a great job so far. Want to zoom in?
Yeah, I was just, when you were going past, I thought if I had a quick look, I'd know whether it's one of the ones that we wanted to sample or not.
Yeah,
Oh, well, that's not, that's a different species again. We've got a couple there, haven't we? Yeah. Oh, at least one of those is the right, two of those are the right species. Oh, no, at least one of them, so that's okay. We can come back there later. Oh, didn't notice. I was looking at corals. Underneath the sponge, bottom red.
Under those corals? Yeah, they're just under that ledge. Oh, okay, yeah, he means
Oh,
under the ledge.
oh,
of that.
there,
Yeah,
there.
yes.
Okay, I see what you mean. I'm with you. Probably an ascidian of some sort, I would say. It does look like eyeballs, doesn't it? I had my coral goggles on. Couldn't see anything else but corals. You do tend to get tunnel vision when you really focused on the study you're trying to complete and the species that you're looking for, and sometimes don't notice all the interesting stuff. So it's so good to have extra sets of eyes to notice the other things that, like fish and sponges, yeah, and stomatopods.
Got one just below me here as well.
There's another big one. Can we zoom you up?
Am I right to zoom in on that? Have a quick look.
Zoom in on one there.
Oh, on the coral while you're going past.
Oh, no, it's fine.
That looks like it.
Yep, those looks good. So we'll get another couple of the Leptoceros while we're here. When you're finished,
That's the color changing one.
Leptoceros.
Have you noticed?
Yeah, you should hear me. It's cool.
We'll see if this one does a.
It's beautiful.
Oh, there's one down there as well.
Yeah, yeah. We'll need a couple more passes after this, if that's okay, Karen.
Yeah, by all means, let's do a few. It's just pretty to look at it.
It's the only thing we've done before, so we just go top of a straight line. Just coming up on the full circle, you can see the traction. This is great. Just kind of a couple passes to fill in the middle. Yeah.
When you're doing the. Oh, he's gone. I was going to ask him a question.
skid count. Is it half like 2 and a half millibar?
Yes.
Is
More
that
like
either more light or less? Just
go this walking like that.
yeah, that's way better. Yeah, that's awesome. That's good.
Go a little bit. Oh yeah, because we're off.
Yeah, there's some big sponges that I
Yeah. I know.
if you run into, but I can only do. Some of them are like a meter and a half long. It's crazy.
So Ian, when you're doing the 3D reconstruction, how do you deal with all the fish in the images?
Well, we ideally don't have them in the images, so it would be the easiest now. So I am where I am at the moment using where I can images without fish, and where they are in things that we don't want in the image, we're using masking. So I'm masking out things that are mobile and maybe some blemishes in the images to help with the reconstruction process.
Just looking at all the little fish swimming around there, you're going to be doing a lot of masking out.
Yeah, so I'm not sure how
Quite a
this one will go.
challenge.
It'll be a good test for some automatic masking that working on in the background with colleagues. Collaborated with some interesting group from Germany where they've been definitely experimenting with masking. It's certainly very useful in the underwater environment because there's a lot of the time there's things moving around which aren't stationary, which can confuse the model. While we're surveying, I think I'd like to just give a shout out to Obertha Primary School in Perth, Western Australia. Particularly, my son's class is Owen in T1 and Luke in PP1. I hope you're enjoying the show, and and everyone to Obertha.
Thanks, Ian.
I feel like you should have been more minutes.
So Ian, yes. So I've done one full circle.
Yeah.
I can do a wider circle.
I would like maybe one through the middle and back.
Yeah, so if you look at my tractor, this is kind of the one. I did one kind of through the middle and
I
go
see
one
it
back.
wider,
But I
yeah.
can do another one in between those two lines.
That would be good, and then I think hopefully we might have enough. I like to see things from the different orientations, like these big sponges. You really need to be able to have image them from all sides, otherwise they're basically going to have a wall missing to their sponge, with a wall missing, one side missing from the sponge.
I can definitely do a couple more passes on it to kind of get the other. And the other thing is, if I come between them, you'll get a better image too. So I'm
That
coming
would be
over them, and you
wonderful
can see
if
where
time
almost
allows. I realise there's a lot to be sampled, but I appreciate the time to be able to try and out some of this reconstruction on such a challenging area, especially with all these fish. I'm going to spend my time masking fish.
Fish and chips.
We're doing that one again, are we? Fish and chops.
It's the coral's still there, it hasn't moved yet.
Mind you, I'm getting dizzy now, so I have no idea which was the one that I looked at before and that we were going to sample.
You look at that one. Yeah, those ones.
It's going well. I don't want to tempt fate, but I think the ROV's providing a little bit of avoidance by the time the cameras are pointing more down than the forward-looking video.
Okay.
But keep doing what you're doing. It's good.
I'll do another couple passes here and get the spiders for that.
It feels appropriate that we do a good job with the coral party to try and what I'm hoping at the end of this is to provide a visualization, so for those aren't able to dive down to what are we now, 50 meters, we're able to kind of fully visualize what this area looks like. Also be kind of useful for monitoring. It's a good reference point, so if we come back here we'll be able to see what's changed.
I just want to 3D print it.
Yeah, on board already. The crew here and J-Rod's already printed off some sponges. Busy printing you a coral at the moment, and yeah, in 3D. Pretty impressive.
Is that one of your corals right there, Karen?
Oh yes,
Where's that, Karen? Which one? Oh, yeah, what's
right next to that sponge.
I was drawn in by the sponge.
I want to pick up one of the switches. Oh, the one's just already falling over.
Looks like there's another one there too. Come back across to this side and probably collect a couple.
It really is a coral party, huh?
There's more small colonies here, whereas the other coral party was fewer, bigger colonies.
Yeah, you're right. Straight right, stitch that up right there.
Yeah. It's just a glad to be there to shoot that.
It should be put right at that. According to your track, like, oh, it's pretty much good. It's a gap for it.
It's going to be by the sponge.
Yeah, I get it.
Yeah, you're jumping over them. It'd be nice to go over the top of that.
Sea snake.
Yeah. Woohoo!
And another one.
Good spot.
You're gonna have to mask that one out of your images, Ian. Oh, he's gone again. Oh, nicely done. Oh, damn! I was hoping to get his head. There he is.
I would like to.
I think someone is giving them both.
You guys didn't want to see where you were going, did you? Sorry.
No, you're not.
Did we get a snick on that one, by the way?
Yeah. Nice to get a fish is one thing, but a sea snake.
I know. That's going to be a challenge for you. There you
So,
go.
one of the few times you don't want to see a sea snake.
The other time is when you're in the water.
They're the same. Same way.
There's another coral for you, Karen.
Oh, where's the coral?
Karen?
Oh yeah. Yeah. What? Sorry. The one in front.
Yeah. This one's fine.
It looks like it. Yep. Oh, it's a lepto. We're just doing a bit of a fly around this for Ian's through.
Yeah.
So what's the frequency of your shots
Yeah.
every second?
Every
Oh,
5
but you can see
seconds.
the stuff.
So I'll just go through this.
Yeah, just the
Yeah.
camera. Yeah,
Oh,
yeah.
that's
So it's good to have.
You just can't see anything.
Well, I think we'll probably be able to get another 5 or 6 leptos here, so then we'll have lepto ticked off. We've got minimum packing cirrus, so if we maybe see another one on our travels, I'd probably be inclined to grab that, just so that there's we've got 3. But you have 1 extra in your bag. We saw another sea cucumber, but we just grabbed one. I think it was more like I didn't actually look. I think it was like the first one. So that maybe buy boxes of full. Needless to say.
Yeah, and then come back from the other direction.
Sure. Just a little bit to the east.
Yeah. Yeah, just need one more pass back through the main two. But yeah, let's go down to the
quite wonderfully magnificent
soothing.
sponges.
Yeah, do we do a full circle around this?
Yeah, why don't we do that? That's all right.
Oh, wow.
You said they were kind of looking straight down almost.
About 40 degrees. If we go a little bit lower.
Texture.
I think it's a coral. It's just the angle.
So it's a street wall.
Yeah, it's a great idea if you could walk around these sponges.
Some people can do
These would make a very impressive. Yeah, if you can get around.
So we
You
have
need
to
enough
stay
angles on it to get to the roots of. It's hard when they go narrow at the bottom for the software to recognise that. So that's when some manual intervention needs to be done. But it's best if we can get photos.
So you can add in our photos, right?
Yeah, we can try and add in. So one of you, if you're able to with a camera with 4K, zoom in on the bottom of it.
Because this is taken every 5 seconds.
Yeah. So I think that's a good idea, Joe. We're trying to see if we can integrate some of the video to supplement where we might have missed some coverage. Just a few fish in those these things. That's great. Are you finding more corals at the same time, Karen?
I found enough to finish my collections in this spot, so it's all good.
Almost enough to keep going for some time.
I'm sure that as soon as we stop moving about, there'll suddenly be a lot more that are obvious.
I'll just do a straight fly over here as
Yes,
well.
please, that would be great.
Yeah, absolutely.
Once you've done the full one.
Where we started, I think. Just down there over the top.
I'm getting dizzy. A little bit.
So it doesn't start that bad.
The final thing I need is to go is the line you've just done to do it in the opposite direction.
Yeah, sure.
And then I think I should be alright.
So long as it's quiver sized.
Maybe
If in
sponge
doubt,
game.
it's a sponge.
When something sees it becomes a sponge.
There's another one of my beasties. So I think we've actually seen that one already. I love the sponge. It's just going good for the darling. No, I think it's a sponge. It's actually got really drippy lips when you look at it from the side. But I haven't looked at it closely, so, you know.
That's a nice mark. That's a nice mark. That's a good one. That's a double one. They hold a nice amount of teal.
No.
I can see the hang.
To explain what we're going to try and do. To explain what we're going to try and do.
Oh, with the sponges. Yeah, cool. We've just got Ellie Hannon here, our artist in residence or artist at sea, and she's had some great ideas of things to do. Great inspiration from these sponges, so I'm going to let her explain it to you.
Yes, I've become a little bit obsessed with these sponges while I've been here. They're so stunning. Not just the colours, just the formations, and they're all so unique. But we're circulating these sponges because we're trying to capture 360 degrees of photos of them so we can create a 3D image that we're going to use to print the negative of on the 3D printer, and then hopefully I can make a plaster mold from that, and I can start making some slip casts to make some beautiful vessels that might be mugs or cups or tea kind of chalices.
Functional and gorgeous vessels, yeah.
Yeah.
Sponge goblets.
Sponge goblets.
Cereal bowl.
Cereal bowls with
Divide that.
Yeah, we wanted to see a gap here.
Very fancy sponge.
I mean, I can spend all day doing this, but I realise we're going to start sampling. It's a really interesting area. It'd be great, Ellie, if you could. It'd be really interesting to take, one of these sponges and do something really artistic with that.
Yeah.
Can't wait to see.
Yeah, it was so beautiful. That one looks almost like an upside down sponge.
But it is actually the right way around.
I know.
That's one of the ones I was trying to describe to you yesterday that has
The
those
different ridges.
straight ridges down it, but it has the little kind of narrowing at the top. Really lovely texture as well. Yeah.
I could make a 4-piece mold for those.
It's only harmless bringing back to the boat.
It could be the milk jug and the other one could be the tea set.
It's just about how much J roll you can make the prototypes.
Yeah, true.
Sometimes when you do this, when you've got things like this, I think you've seen it before, but I'm just wondering if
I just don't know if that's possible with your ROVs.
anything.
I like that on here. This I love that attitude. I don't know if you heard that anything is possible, and that's just what's it's very indicative of the wonderful philosophy on the Falkor. Impossible is just a challenge.
Challenges are always accepted. See, like that one right there.
All right.
It's not attached to the seafloor. I can grab the bottom and I can spin it around.
And there's a great place to land right beside it. It's like
Yeah.
Yeah, okay. It's got to be done, doesn't it?
Sorry, Karen.
It's got to be done.
Yeah.
I'm all for it, absolutely.
That area that I divided in half.
I think it would be the first to be interested in a Is it? Would it be a
I'm
vase
getting the
then?
first
Chalice.
teacup.
Oh, is it? Yeah, a teacup. Wow, that would be a lot of tea.
Well, I have.
Could we fill in that hole in that one there? Could be a problem.
Especially with the tea drink.
Maybe that's where you put the handle.
Yeah, that's true.
Oh yeah, I was thinking about putting a straw there, but I don't think you drink tea with a straw.
No,
Well, not if you're British.
not if you're British. Drink it out of the saucer.
This is where science meets art. It's an amazing space.
Yeah, it's very
Hey,
exciting.
just so you're aware, I
Yeah.
divided those two lines, but it was fairly sandy in there. So
Yeah.
I think we did get pretty good coverage on your inside.
Yeah, okay,
there.
well that's always good. Thanks for letting me know, because I think that's one way to know if you've hit the edge.
Yeah.
So here we go, we're going to probably the world's biggest, possibly first sponge and spine tea. Is it teacup? How do you haven't decided yet, I guess?
It's going to be used for everything. Whiskey.
I'm just going to see if you decide, and if not, we can do more
Milkshakes.
video. Okay. Yeah, so milkshakes.
I reckon an ice cream cup would be good.
Cream. That would be
Yeah.
good.
Nice ice cream flavour.
Wow, they're pretty solid.
Oh yeah. They're heavy as well.
They're heavy. Yeah.
You can have the handle as like a sequence.
Yeah, I want to pick it up and I'm grab it.
And like blend it into sort of like camouflage in the same
Serendipity that it was just sitting there. I think it was just waiting for us.
Do you think you just need one arm to pick it up, or maybe two?
What's that?
That's right, you focus.
Looks like it's going too hard, doesn't it?
I didn't grab all the way down. I didn't know that.
Oh, you've got two arms.
Contract this one. It won't fit
I
here.
mean, one thing I could always do is
Both arms.
yeah,
And you
definitely
can't
go
spin
to a
it.
dump.
Because that's about 2
That's
hours.
great.
We just bring it on board.
No.
I don't think it'll fit in the bio box.
One thing I could do is I could just rotate it around, right?
Yeah,
And
I was
then-
wondering that.
And then your camera would.
Yeah, we could try it. It would be experimental. I'm not sure how well we'll go for that, but we can try.
I could definitely pick it up with two arms.
What about this, Jared? Could you get at least on the bottom if you got it in your hand?
So use it stationary time points where we'll kind of attempt the bit where you explain that it's not moving. Yeah, sorry, it is moving and you're not moving.
So you need more.
Yeah, I'm not sure it's going to, but I'm happy to give it a go. Let's try this thing.
Or if you turn it upside down, but then you won't get the move like the top of it.
Can you? Yeah. Can you? Can you go around it while it's one way and then turn it upside down and go around it the other way?
I could definitely do that.
Yeah, I think that would then move the arm, turn upside down, move the arms out of the way, and then we'll get that nice texture on the outside. And then if nothing else, if we've just got protection on the outside,
We can change the top of it.
Yeah. So
Yeah,
I think
'cause it is
the
the
thing that's
outside
interesting
texture,
is
it's
the outside
great.
texture. And this would be a very delicate operation. It's almost.
I don't know if there's anything else I should be doing.
Oh.
Like playing soccer underwater.
But it's harder.
It's a good effort, but it didn't require.
Just center, yeah.
So it's
This should be.
going to try with the other arm now.
The commentator.
Well, it's where you turn to the pundit and say, oh, you know, it's a good effort by the lad there, but oh. Here we go. What about the fan?
Oh wow.
Wow. With almost surgical precision.
And then you realise you're underwater when it doesn't just quickly fall.
Oh. I think it's that fan, so you're right. Yeah, you move it just a little bit.
Kind of looks like one of those.
So the fans
The old
free.
salon things that you put over your head.
Oh yeah. Well, somebody suggested that Sebastian should wear it as a hat.
But I'm pretty big.
So now it's put it on the camera. So now you have to just spin around.
You do that. Just image around it now.
Yeah.
Lampshades, flower pots, lots of ideas out there.
All right, so that should be in view right there, right, Ian?
Oh, so cute. Oh, like 3D printed.
Yep. Do you want to move him just kind of right in lines? Yeah, and then we can pay out just a little bit.
I'm excited.
Okay, wait one
First one, so can we do one just an inch back? The audio video as well, which is going to help with it. And then I think we should have I think a bit good to the 3D image now. Oh no. You're towards the boat.
Hey, Russ, can you give us 10 meters?
Roger that, 10 meters.
Oh, thanks, man.
Just one more inch back.
Sure. A little bit back.
Yeah.
Yeah, it's probably good there.
All right, then you should be
Further
good
back?
now.
Sure.
Are you going to flip it back over when we get done, Ken?
Yeah. Do they need to do the inside as well? Let's do the inside as well, hey?
Ian, is that better? Just a little bit more back. How's that?
Bit more. These cameras are facing more forward, really, aren't they?
Right there? More.
Maybe just down more.
Problem is there's all those corals on that one side, so I gotta come about this height.
That's good? Just top of the camera.
So cool.
I think I'm good if
Did you need the inside, the top side of
Oh,
it as
yeah, if
well?
you can.
If we turn it back over and do the
if we turn
top.
it back and we'll add the top to it and maybe do a separate line.
Well, though it might not sit upright, it might be a bit harder.
Good for me to go.
I am stable enough.
Wow, 2 arms at once, I'm impressed.
Impressive. Wow. Yeah, if you can put it away from the fan, that would be good.
And we drink over your base.
Now it feels like we're at the beach. We're going to have to build a sandcastle mount. Oh,
That's dedication for you.
So messy. Having to have to
Yep, I think the whole set's being left on board Falkor.
Just find a spot to just plop it down and like just stay.
It's kind of hilly there too, so it needs that.
But is it because it's no longer attached? It'll still be okay, won't it, Karen? What sponges do they? Is the attachment they'll maybe reform or they'll just keep growing?
They should be able to continue
Yeah.
to grow.
It's not like
I mean,
it's
we've
roots
we've
or
seen
something.
quite a lot of them already upside down as
Yeah,
we've been,
just keep getting bigger and bigger.
moving along. So yeah, often the attachment point gets eroded by
Like an
other
extra.
sponges and other organisms, and they get hit by large fish. So yeah, it's a kind of a natural process for things to fall over.
That looks good. That's amazing.
Well done.
Just so impressive with the arms there. I bet they're really good at those when you go to the arcades and there's those grabber arms.
No, those things are designed so no one's any good at them. Sure of it.
Alright, let's go down with it.
How's that, Ian?
I'm so jealous. That's awesome.
You did great.
That's good?
They ask lots of questions at the end. Did you get those?
They asked questions.
Oh, cool.
We kept just doing like a follow response, like if we moved anywhere, we're like, any questions?
Excellent. Oh, that's great.
We could answer most things, but they had some like high tech questions about the ROV that we were Like how does it move and like so about
How they
fiber
get into the
optics.
water.
Yeah. How fast does it go?
I'm impressed.
Yeah. No, Johnny, my ear was like fiber optics. I'm like, ah, yes, fiber optics. For sure. I know what that means.
So good.
Yeah, that's what we're in here.
How does Sebastian know what to do on there?
It looks like it goes there.
The pilots. Our work here is
Yeah, yeah.
It's like cord attached. It's like flying a plane. It's like sitting in it.
Yeah.
Mm.
I kept saying like a remote control.
That's true. Yeah.
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 How did the people go down with it? That was really good. Smart bunch. Yeah. They're classes that I do 10 weeks. It's around 10 weeks of teaching, which is great. And so when I was leaving,
There's good dancing.
It's not the same. For me, you have a whole camera. Thank you very much. I just hope I can, live up to your, do justice of your driving and Eddie's artistic skills. I guess over to me, and I'll go back to the other bit of the lab.
I can't believe you got this one standing up.
It feels more right now that it's in that direction. I guess it's over to Karen and sample time.
Yes, that coral is one of the ones that I'd like to collect. So yeah, when you've finished dancing with the sponge.
I'm good, but enjoy your sampling. Thank you very much, Karen.
No worries. We look forward to seeing the outcomes.
I just hope you can do justice of your coral party.
Party. A coral tea party.
Oh, don't talk to the British about tea parties.
Oh, of course not.
Yes.
I think this is definitely an Alice in Wonderland tea party, though.
So, alright, that's kind of basically back.
Thanks, guys. That's fantastic. Right, back to the science.
See better. Start turning the other way.
Now you've put that sponge in just the wrong spot, haven't you?
What's that over there? Oh, there's that spot.
Oh, we can have a look there, yeah.
Email the guys from the quarter.
Okay.
Or at least maybe just forget.
Ah, so that's the
Yeah. So I was like, I've seen this before.
cell of the Harley sponge. Maybe it is. Oh, just a sponge on that.
Yeah. It's got to be monkey ass back.
Pour around on the truck. Sit down. Just don't rotate too much.
Pour around?
Yeah, cheers.
A little bit off?
Yeah.
All good?
I'll be on. That's perfect.
Which one you want to go with?
I'll get it in a second.
Yeah.
She's gonna look at it first.
Okay.
Are you happy? Yeah, yeah. My turn. an in situ photo if you wouldn't mind. All good? Actually, I'll just do a close up.
Got it.
Thank you. And we'd just like a itty bitty sample of this one, please.
Okay.
Perfect. Not quite perfect.
Another little piece, Karen?
Nope, that is just fine. Thank you.
Now there was that one that was kind of close to the edge, was the long sprawling one. You want to go to that one?
Okay, that sounds like a good spot. Thank you.
This one right here.
That's the big one you like.
Did you get everything? I have to push it after you push this off. I'm just gonna brace it a little bit.
Not a problem.
And this usually has so much use. The rubber needs to be more easier. Help you along. Oh yeah, they're pretty great for that. That is all big.
Sometimes you can use those two electric shear bolts.
Really?
Right, so I use this little ball and put it in the hole.
It's crazy. That's pretty good there.
Happy?
Yeah, it's all good.
Excellent. Thank you, gentlemen. Happily Leptocerus colony. Oh, fish. He wants his photos taken. Could you do it in situ for me, please?
situ.
See if we can get
You just take that other one for the bit more polyp detail. Thank you. And I would just like an itty bitty sample of that one too, please.
Floppies?
Yes, please. Lovely, thank you.
They're just taking that bit of the,
I was just going to say, do you want these other two pieces as well, or not?
they broke off quite nicely. Yeah, if they
I think they'll probably be easy to pick up.
yeah, that sounds good next time.
That whole thing.
Yeah, do you want us to pick up the rest of the
Yeah, I think so. Since they came off, we can use them for the ecophysiology. Don't let them go to waste. That's number 12, isn't it? Was that the one that was meant to change colour? Did it?
I didn't pay attention.
Oh, you mean you were busy doing something? Lovely.
Do they like fuse, like little ones all fused together? Because that had its own little base.
It could be two separate colonies that fused, or it's just actually attached to the substrate as it's kind of grown, if you like.
Just added a new anchor point.
Yeah.
That's good.
We did see some the other day. I don't know whether you were on, but there was two sort of colonies that size, and right where they joined up, met, they were fusing together.
Is that good or?
You happy with that?
That's perfect, thank you. Now, before we move on, I just want to take some photos of this other coral that's in the foreground, because I'm still going to wait for Amy to tell me what genus it is. I'm testing her. We'll get a better look at this one. It's the same as one of the ones from the other spot, but it's easier to see.
That's all you, Karen.
Thank you. No, it's not the same as the other one. I lied. It's different again. Oh no, it is. It's just very fleshy. It's we just need to poke it, and you can see the skeleton a bit better.
Okay.
Yeah, poke it. Sorry, do you want to zoom out so you can see to poke it? Having a competition with an Ascidian there though. Or is it a sponge? One of those things. Can we convince it to draw its polyps back a bit? Oops, poked it a bit hard there. Yeah, I was hoping it was going to do that too, but it's not going to. It makes it much more challenging to identify it when you can't see the outline of the skeleton underneath the tissue.
Since I got this little piece broken, you gonna grab it, have a look?
Why not? Probably a little bit too little.
Oh, that's pretty broken.
Yeah, it's probably
Do you think it looks a little bit left Astria-ish? I can't quite tell if it's going in like that sort of evolution.
That's what when I saw the big one, I thought it might have been something like that, but the polyps are too big for Leptostria. But you're in the right family. Okay, I think we'll stop being distracted and go in search of more Leptoceros. All right, now, can you remember where all those other ones were?
Yeah, there's one just up the ridge on the right hand side.
Okay.
Let's. The big ones or the little ones?
Yeah, there was those bigger ones at the top that we haven't had a good look at yet. Let's go and have a look at those. Because I'm sure that where we find the big ones, there will also be little ones.
That's what the little ones were. Yeah, just big ones.
Oh, so you can find it.
There's that one up there. Wait,
Yeah.
is that one already?
That's
There's a
three.
couple up there, yeah. So I think one of them at least is the right species. I think a couple of them aren't. That's correct.
Do you want to try to sample this?
Probably, I think, yeah. From memory, that one in the back underneath is the Leptoceros.
Okay. Let's,
check with them for a sec. Yep.
What, those three? The three are backloaded?
There they are.
Or maybe quarter.
Was that? Yeah. There's like those that are slashed to the left. If you go like that right there, I think it's
Thank you.
polka dots.
Can you just take a gratuitous photo of that for me? It's very pretty, but it's not the species that we're after. Okay? So, the one underneath here, however, is.
It's good to go.
Yeah, do you want to pick up 5 meters?
Copy.
Cheers.
Could you do the in situ photo just there, please?
Is it pay out?
Now, down that way. It's another species of Pegasus, but not the one that we're sampling. And can you do that whole, so I can see the whole colony shot as well?
Yeah, just because I hate that.
And this is the echinophilia. And that's the echinophilia / oxypora thing.
You can get it there. Yeah, I can get it there. Actually, I can give you one outside.
Oh, actually, before you do that, I might just, because this is another Leptocerus there by the looks of it, let me just zoom in on it. Oops, up, down. Maybe by day 14 I'll have worked out the right way to drive this thing.
Thanks, Greg.
Yep, so that's a Leptocerus there as well. So we'll just... Can you also do an in situ of that? And so we will sample just a small amount from that colony and a small amount from somewhere that's easy to get to from that colony. How's that?
Different corals?
They're two different corals, so two different quivers.
You can almost even get it in the right there.
Yeah, you already got that.
It's that bushy thing. Looks like black colour actually. I'll have a closer look at it. It's not as white though, it's a different colour. Yeah. Perfect, thank you.
So we'll just straight off the bat, this whole area right here, it's like coral party. So we're just kind of searching around. Okay. Some corals. That's pretty much it.
Of course it landed in the hole. This is where you find it holding your breath, going, is they going to get it? Woohoo!
Well done.
Superb sampling there by J-Rod.
Okay. Do you have
15. Sweet, perfect time.
we'll put it in there.
I got one there, just bring the camera back. I got great sort of bonus.
Just taking a photo of that, I reckon.
Just bring this down.
eventually. I bet if you'd tried to do that, you would have been able to. Beautiful. Now, before we scooch too far, there is a black coral there that we'd just like to have a little squiz at.
It's all new orange.
It does look a bit like the one yesterday, but a bit bushier actually.
Nice.
Let's have a look where we can see a bit more daylight through it.
Thank you.
It's certainly got a lot of similarities with one we saw yesterday down at about 160 metres. Oh, I see a little shrimp in there. Oh, there's somebody else hanging around in there, a little crab in there as well. Let's get the size on that. Could you put the lasers on for me? I just want to... Oh, are they? They're hiding somewhere. Oh, there they are. There's one, there's two. Is that... It's, yeah. So it could just be squidged in a quiver, but it would be squidged. Oops.
Yes, so Jeremy has just identified this as Cupressopathes, different species than the big one we found yesterday, but the same genus.
Yeah.
No, I actually use Schmidt, so they I just pick this out.
Just trying to not get the quiver in the bottom.
That's good.
Could we take a photo of that, please? You got that? Thank you. Okay, I've looked at that, Oh, I'm just going to have a quick look over here. And then right, I think I'm done with that spot. Oh, you want that?
Oh, yes.
Oh, you
Oh,
want
yes.
it?
Oh, yes,
Oh,
yes.
you want
I was
it?
wondering why you were getting distracted.
I thought you just wanted a photo
Oh,
of
no,
it.
no. No, 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 no.
No always trust the museum folks to want to collect something. Can we collect that, please?
The leafy one.
The big, this big thing here. It's a black coral. We'll need to take it from the base, please.
Number 10 there, Ben, if you can trolley out a little bit.
Oh yeah.
Actually, that's not going to be easy. You wrap the camera
It's,
back, you know?
Yep.
See if I can get it from there. It's enough. Come bring the camera to that stuff. It's in there. Let's see if we go.
Which one you going for?
You know what? I was going to go 10. Okay.
So let me pull the camera back.
Yeah, please, as far as you can. Should play that one. Come on, chill out.
Did you know there are ROV that specialise on black corals?
Oh, no, I did not.
It's really curious. They cluster up to sleep at the base altogether. So I'm interested always to look at the base, but also
Not just for the taxonomy.
Yeah, yeah. But I haven't really been looking at the coral that closely. But for some reason, I've sort of forgotten that there were some specialised on things.
There's only so much you can remember.
Yeah, now I have a double interest.
It's not full.
So from here, we could either wander to the more shelved side or the steeper side. What's your preference?
I wouldn't mind just doing some close looking upward.
Yeah, shelved side is kind of easier to sit on. Once we get to the steeper side, we've got to kind of, unless there's a,
Yeah.
we should still look, but
But even just up to the top is interesting. When it's
order.
Okay, let me put you in a little bit.
yes, yes. Also like very real day.
Which is a terrible shot.
Use some sit.
Yeah, please.
Zoom in when you get closer.
Is that okay there?
Yeah, yeah. I mean, the very base is fantastic if you can, but if you can't, no big deal.
Okay, right at the bottom.
Yeah, that looks good. Perfect.
Perfect. Look at that.
Get back in. You know. Some shit.
Fabulous.
Well, I guess there's one current should fly. Should go straight down.
I ideally you want it bottom down.
I think that rock will help draw it down, straight down.
Okay.
So I think,
I was
yeah,
worried about damaging the top and put the plunger in.
that's okay. We can, it's a little bit flexible.
It's not heavy enough.
Okay. Which bio box shall we try and squidge it in?
I'll put it in a buy box instead.
Bio boxes are technically quite full, but
Probably the only
actually we
one is
could
the
probably
one.
put it in G left because the two colonies in there are fairly small.
I think it's too long. Do you think it will fit across on you, the big one?
I mean, that's your size. Let's look at it pan down there. I mean, I think diagonally it would fit 1/2.
Okay.
You know what I mean? If it goes diagonally across the water, I think there's enough room for that.
Okay.
If we go on the left side, that makes it easier.
Yeah.
That's probably good enough there then.
Are you happy for it to go on the left one, Karen?
Yeah, the two colonies that are in there are both be fine with that in there.
You gonna put it here?
Just open up the left hand side, because I can. Because if this falls out, that's fine.
Yeah, the left one's fine.
Very nice. Let me move the card, because that card's going to fall in the box. Guaranteed.
Yeah, porch in a little bit.
Yeah, I got it. Watch it.
I'll watch your base. Still clear. So. Nice.
Okay, there's it.
Okay, watching out.
Okay. Yeah, box. So keep going, keep going. Keep going. Just got more left. Got a bit more.
That's it.
It's not bad. Okay, you can hand me down.
Let's porch out a bit more.
I get down so cold.
I'll drop in there.
You just hand down.
Bit taller.
stuck in there then.
Closing. Watching.
I know we should let
Camera's ours.
All right. We good? I think we can move to another spot. Let's. Oh, you want to just do a general scan here, or okay? Do you want to swap chairs? I'm going to hand over to Nerida now. We have technically collected all the corals we need to, but I'm sure I'll convince her to find another couple on the way. But yeah, we're going to start doing a lot more searching in detail for mollusks and small critters. More fun to be had.
Left off from here.
No, not yet. Thank you.
Nerida gave me that look.
No, there's more fun to be had here.
Turtle on the aft deck. Roger that.
Not on the aft deck. It's in the water.
Roger that. Apparently we've got a turtle on the port side aft deck. Here you go.
Turtle alert.
Yeah.
Turtle alert.
Be nice to get it on the aft deck though.
Oh, you have to go.
Say Ben's in there, he can probably get excited.
He's already thinking about soup. I had conch once. It's very good. I was in the Bahamas. And well, we've had a lot of them because that's like.
zoom. Let me zoom out.
So you can adjust it.
See where it is. Oh, is that nice? It's a side level.
Oh, sorry, I've just seen it adjust right.
You want a piece of this one?
Now you've got something in there. There's something I see there.
Is that what you want?
It looks like a.
I'm just trying to focus on it so I can actually
yeah,
see.
just-
Yeah. Just the autofocus doesn't seem to.
Let me see. Try that. That's almost this way. Clockwise. Right.
Oh,
I think it's right at the end of the zoom.
let me try counterclockwise.
It's focusing past it.
Zoom back a wee bit.
Go ahead. Yeah. There, it's good.
Go back in a little bit at a time. Just try refocus. Right, refocus now.
Yeah.
Because it's easier to do it incrementally with the manual.
Try a little closer. Hold on. Let me get a little closer.
Still stop there, Ben? I'll stop. Let me do it now. Okay. Yeah, we're out of range. We need to get into that one. It's not that focus.
Let me try it. Is that in focus?
No.
You're at the end of the camera's
Yeah.
zoom, so it's not able to do it. You'd have to get a little bit closer. Where is it? Where in relation to is it? Are we looking at?
Yeah, it's right in front of us.
Really?
Yeah.
Oh, yeah. So it's,
Just one of those.
I'm gonna do it by myself.
Are you in autofocus over there?
It's off here, but it's just not.
It's focusing on this little guy behind the. Oh, there we go. Whatever you just did then, now it's changing. It's gone past.
Maybe focus in the face. It's thicker.
I'll give it a try. So,
Sorry, folks. Sometimes this gets a little difficult.
I'm gonna zoom out a bit. What's that one? You want to zoom in a little bit? Is that in focus?
Yeah. Look, I don't think I can see anything interesting enough to probably persist with. I can see enough now, I think.
Yeah, the camera's yours.
Yeah, cheers. I think we're ready to
Take off.
hop along.
Is there any particular direction you want to go to?
Not really. I'm interested in looking at just fans, some of these. Like some of these ones like this would be good. Awesome.
Okay.
Thanks, man.
It's kind of a routine. You're sitting on a.
Yeah. Let me back up a little bit.
Okay.
Like you slide over.
There you go. I'm off the hill.
Do we have to make that go back on autofocus again, or
Say again?
do we have to put this back on autofocus again, or
I just put it back.
Is that all? Yeah.
these whips going to be challenging?
Zoom in slow.
Yeah, I don't think it had it in the first
Yeah, you
place.
can have to use manual for it. It's just,
Yeah,
it's just the way it's.
not enough of it. Yeah.
Okay,
And so is that better to be close up to start with, or half
yeah,
and
you
half?
should be able to wait in the back. You just wait that right there. I'm sure why it's not. See if I can adjust the weight a bit. Can you try that maybe?
With the auto again? Or
Try, no, hold on. You go to base.
It's like they're all forces just not working. Here. Yeah, usually you have to reset it and then go back. I don't understand why it's not working. It's not set. Just adjust it. It just takes forever. You can wind it like 8 times. Okay, you can see them. They're clear now.
Hope this isn't making you seasick. Quite a moving target.
The back ones are in focus. That's healthy. You want to first get some salt?
Oh, cheers. Wow, I think I should have taken up cryptic crosswords instead. How do you then, after that, how do you then make it go back into the auto mode?
Yeah, you just press it. That's the auto mode. So this one here.
So it is on? Oh, okay.
You'll see
okay,
it when you press that.
Right.
If you look at the screen up here, you'll see. Oh, that's good. It's off. This one. Anyway, we
Oh,
can
yeah.
adjust it back and
Gotcha.
forth. So
I see.
it's on.
Thank you.
Some bottom bag of those. Seems to have a lot. About 2 months worth. That must be 4 weeks. I don't eat 3 so
is in order, but it's not actually focusing. I don't really understand.
If it's close, it can't, there's too many things. It's not focusing.
Okay, now it is though. But it's it's weird.
They just go in and out of auto. Is it in focus?
Yeah.
I can't judge in focus.
That's what I was thinking.
That's why I asked her, is it in focus? I won't trust myself. Yeah, if it's in focus. Right? That one you might have to do it manually.
Yeah, and it's far, it's okay.
It's like in focus.
That's good. Alright. So can we take a look at one of these kind of ones?
It's right behind us, in front of us.
Yeah.